is that?
Big shrimp.
Behind it.
Little fish.
I know, but what kind of fish? Oh my God. Everyone's a smart. His guts. What is going on there?
Good zooms on this guy.
Okay. All right, so good morning. We just got to the bottom. We are just to the north of Tobago, starting today at just over 750 meters. And wow, that is cool. Trying to identify this translucent, weird telescope fish looking critter. He's gone. All right, that was cool. Oh, there he is again. Oh, making a comeback. Okay. He has two fins. Yeah. Wild. Yeah. So is it just his head that's translucent, or the whole? No. Oh, yeah. Wow. Cool. So we just got to the bottom, and this is the first. Well, we got dusted by a squid on the way down, but this is the first crazy looking animal. Look at the.
He's got upward eyes.
Yeah, upward eyes, completely.
It looks like one of those kids' science things where you can see the skeleton in the body.
Yeah. Oh my gosh. Wait, what did you call it? Nothing.
Juliana's just making things up.
Yep, no, I know.
Alright, I got it.
All right, that is wild.
I think it's probably larval of some sort.
Okay. Cool. All right. So we did lots of mapping last night. Oop, shrimp. Trying to find a place where we could both encounter steeper slopes, so potential coral gardens, as well as a place where we could dive safely. There are just wicked currents around here. What is that one doing?
Same guy?
Is it the same one? He is totally dancing. Yeah. Wow. What is that behavior? That's so funny. Okay.
You can do it again.
I'll do it again.
Ready?
Whoa. The sound effects are the best. Bing. Yep. Wow. I mean, he's like pogoing. Oh, that's so cool. Again, one more time. Yeah. I mean, maybe he's getting tired. It's like, I need to. Oh, is that the same one? Or no, this is a different fish. Okay.
There are many fish.
Yeah. Oh. Yeah, he's clean. He's curling. Oh. Woo. Pogo fish, yeah. All right. No one knows what that is, but we'll get back to you. Wow, look how silver he is, too. Beautiful. Wow. Metallic. All right, so this is Rachel Lauer from the University of Calgary. We are just north of the northeast part of Tobago. After spending, I think we've done like 16 hours of mapping maybe so far, trying to map this area that sort of wraps around the northeast of Tobago, which is a UNESCO site. And had previously, we've been relying on these Gebco maps, which are very poorly constrained.
Up to
the first thing we're going to do, this will be our, I mean, we're planning to do one more dive before we head back to port and get off the ship after a month at sea. And this is our last biogeographic region that we will be exploring. This one is a bit shallower than we have been. Today we're starting at around 750 meters water depth, and then we will be moving up to about, what, 500 to, and then 300, yeah. So we'll be ascending from here. And between now and then, when we hit that cliff, we will be starting by doing a transect, so a biological transect. And then we will head to shallower region where we hope to find coral gardens and sponges and other critters that like being on in areas with high currents and also steep slopes. And that's primarily because in steeper slopes you also tend to have less sediment, so more exposure for rocks and things to attach to. But we'll see. It's hard to say by looking at the bathymetry whether something is hard or sedimented. What's that? Oh. Hey, there's is that a halosaur? To the left. I know those guys.
You're like, you knew that one.
Well, I know. I've learned stuff on this cruise, guys. Not as much as I should have. Juliana, this isn't vertically exaggerated, is it?
No.
No.
There's just one right now.
Yep, that's what I thought. Somebody was asking, I was like, I think that's, yeah, it's just that.
Normally
Anna, what
we
are
do
these?
have these at 2.
Anna,
Yeah,
This is like,
yeah.
Anna, what are these things?
The red. What was it?
Okay. Yeah, there's one coming up here.
What is it? Oh, sea pen.
Sea pen.
Yep. The little, it looks like a little truffula tree.
No, you mean the thing? No, it's a stock rhino.
Crinoid.
Stocked.
Stocked.
That is a stock.
Okay.
Mhm. Hmm. What are these guys? Just big shrimp hanging out? Yeah. Yeah. Just a couple of shrimp. Ginormous shrimp. Having a combo. So, are these sea pens? This, or is that what you're calling a crinoid? Stalked crinoid? The white? Yeah. Got it. Lots of them.
Another one that's sticking out on the left there, that's the sea.
Okay.
Let's get around here.
What's the water temperature? Where's that? 5.8. Oh, is that a lizard fish? Maybe. And what is that? Oh, okay. Anemone. It's okay. Yeah.
Some kind of weird fish.
What kind of fish is this ahead? It's swimming in front of us?
Which one?
He's moving like that way. He was hanging out and then he whoop. Yeah, I don't know what that is. Like something crashed.
Yeah. Some kind of fish with big, huge fins.
Yeah. Is that up here? Yeah. We
Our sponge on it.
yeah, should we
There it is.
check that out a little bit?
No.
No, keep
It's
going.
sargassum and we have the sponge.
Okay. We have that
We
one.
have that
Yep.
sponge.
That is a beautiful sponge. It's a glass sponge. Yeah. Okay. So
Transect?
Yeah, just slow. Don't land. Just as long as we stay on the transect.
ooh, there's another beautiful stalked whatever. I know, but I'm trying to remember which one it is. Don't tell me. Hyalonema. Yes. All right. Hyalonema. Yep. Oh, there he is again. Oh no, different one. This guy. That's a different. They look cool with the shadows.
Oh, no, it's not. It's a fish.
This? It's an urchin. Yeah. Chunky one. So at the moment we are on a transect that's parallel to the slope that we will be ascending later on in the dive. So we're staying, basically following a contour line. Oh, cool. Brittle star wrapped around, what is that? Is it just sargassum? No?
No, we're trying to get pictures.
Huh. Well, the crinode's on the right, but what is the brittle star wrapped around? Piece of wood? Cool. Yeah. Tiny wood fall. Nice. Oop.
From this area, that fish could be brown snout
Is
sweetfish.
it?
No, the first one we saw with the eyes.
Yeah.
Clear guy.
Oh yeah.
They're in the area.
Cool.
But,
The eyes were really close together.
yeah. When they do this, they're like pointing up.
Oh yeah. They
had
look
weird
a bit
little
like
round
aliens.
snout as well.
Oops, something up ahead. Looks like. What is that? Crab? What? Oh, it's a lobster. Why is his tail up like that? What is it? Yeah.
Problem with transect, I guess.
We could stop and, as long as we stand in transit, we can stop and take a look.
So I just want to get a nice.
It seems to be online.
Very dusty. Lots of snow. Those are some big claws, huh?
Look how sharp hands on them.
Yeah.
I gotta stop the boat if I hang out too much.
You want to stay here long or just?
Well, I'm hoping somebody's gonna tell us something.
We'll have to stop the boat if we want to.
No, that's fine.
No, we're good.
I don't want to respond, but I want to respond.
Whatever it is, I don't think we've seen it before.
Weird. Super weird.
What do you say? What do you say? Like, oh, crayfish? Oops. Give a picture.
Open, yeah, 'cause the claws are quite unique on them. Right?
Yeah.
They were like giant claws.
Point. Your person claws. Interesting stuff.
Mm-hmm.
Be excited to see this rocky area.
I know. I just hope it's rocky.
And not just
Yeah. Exactly.
I mean, it's pretty steep, right?
It is. It's not.
Maybe.
It's not an insignificant slope. I would say it would be the steepest one we've come upon.
Okay.
It's something. Looks like another Hylanema on the left over there. Good morning to those who are just joining us. We are on our second dive in the UNESCO area of Northeast Tobago. This one's a bit deeper than our previous dive. We're starting around 750 and whoa, octopus. Okay.
Okay.
Wow. Okay. In,
It's perfectly sitting.
is he in the fight position or is this
Position we find.
just the position? All right. So,
So,
uh,
keep going or hang out?
Sorry.
it is a male.
Male moose octopus.
Okay.
Hold on.
You wanna pause the boat move there?
Yeah. So, this is the.
ROV, can you stop the boat?
Oh,
Stopping now.
I don't know what the next expedition is. Do you guys know?
The next one is like, it's like gliders or something.
Gliders?
It's not our,
It will not be an ROV mission. So no, obviously not as cool as this one. Wow.
Oh, buddy.
Oh, buddy. Huh. Yeah.
You haven't had a responsibility. Hold on. No, all good. Oh, no,
All
sorry.
good.
No. That was not from Diva, that was from
No.
somebody else.
Alright, still two.
No, it looks a bit rough.
Yeah.
So,
Yeah, so we'll just collect some
Good
imagery,
imagery.
but we don't need, yeah, that's it. He looks like he's been in a fight.
okay. Yeah, just good imagery, please.
Hmm. Wow. The pattern it leaves. So this is a male moose octopus. Oh.
Oh, there's our friend.
Yeah, it looks like he's got some scars, battle scars. Just maybe they won't notice if I just go over here.
Exactly what you do his
Yes.
Uh,
I mean, his nose is pointed towards us.
yeah.
Look at those weird circles on there. It's like,
Yeah, what is going on? I know, that's, yeah. He's been through something.
been through it. We all, buddy.
We all have, yeah. Well, yeah. He would be like, what is wrong with these people? Mm-hmm. Oh, he's beautiful. Wow. He's tired. I'm gonna take a break here. Yeah. Perfect. You wanted to zoom me on the crinoid, right?
These are exactly.
Okay.
This is a nice shot.
Beautiful. Yeah, he's just,
Like a lady.
yeah, he's just on a stroll. Just morning stroll. Doing a little yoga, stretching.
That's great. I think we can go.
Oh,
Yeah.
cool. Look at the marks he leaves. You can see the, yeah, it's cool. Now we know what that is. That's so cool.
See you, buddy.
3 to go. Mrs.
I thought she
Very
was here. What?
She was here.
She's so sneaky.
Care about nothing but octopus.
I know. Lots of people from Uruguay here.
Resume or move.
Sorry, Gerald Beach. It's good.
We are for those just joining, we are at the end of 30 days of diving in 93% of Trinidad and Tobago, which is waters deeper than 200 meters. And today's dive is just northeast of Tobago, where we are diving in this sort of horseshoe-shaped area that is a UNESCO World Heritage Site. And previously, it's been, a lot of scuba work has been done, but no ROV work has been done. And we've spent the past couple days just mapping as much of it as we can, because the maps that we have are very coarse and they're based on satellite altimetry data, which is just gravity data, which is notoriously bad in terms of resolving anything, any real structure. So we've been using the multi-beam, the ship-based multi-beam, to map this horseshoe-shaped area as best we can, doing the deeper areas at night and then the shallower parts during the day. Oh, that's beautiful. There's a little squat lobster on the glass sponge. Halosaur. Yeah, we've got a lot of sediment in the water column. We are close to land, but we're also in a pinch point for a lot of ocean currents that kind of come together just north and south of Tobago. So the currents here are quite strong, which makes it a challenge to try and find a spot where we can dive. So you don't want to spend all of our time fighting currents. We'd like to be able to stop and check things out. So a lot of decision making was tied to that in terms of where we were going to dive today. So I'm gonna share some of the cool research I was doing about this site. I mean, in general, this area is part of what's called the Man and the Biosphere program, which is an initiative that was launched by UNESCO in the early 70s to try and balance the protection of nature and acknowledging that humans are a part of that. So its core values and goals are conservation, sustainable development, and education. And this area is, in particular, was designated in 2020. And it includes, part of it is also on land. And that part on land includes this, the forest reserve that has been protected since 1776, which is crazy. And it is the oldest legal tropical forest reserve on the planet. Yeah, so the idea is that Tobago can protect this really unique area from exploitation and responsibly steward this area for the benefit of not just future people of the Trinidad and Tobago region, but in a larger scale, just thinking about the scope of preservation that, this is a model for that sort of work. What is that? Is that a... Yeah. Is that just a... Yeah, I don't know what it is. There's something up ahead, crazy looking. That is... I don't know what that is. Huh. It looks like wood that is encrusted with critters and little sponges and anemone. Oh, cool. Nope. Yeah. And some brittle stars wrapped around it. Cool. So we've got several examples of woodfall so far on this transect. Habitat. Yep. Maybe another one? Another tree stump looking thingy. It's always weird to see, yeah. Similar. Yep. Nice. Ooh, look at you. That's pretty, bluefish. I feel like a Dr. Seuss book every time I'm sitting up here. Cool. Sargassum and treefall. Cool. Yeah, there's quite a bit of woody debris. Maybe we're near a river delta or something. What's that? Oh, that's another, yeah. It's all right. We're good. Anemone. I couldn't say that word until like 2 years ago. Anemone. Another 100 meters. Okay. And so we're gonna go 200 more and then come back to the waypoint. Make a left. Or sorry, right. Your other left. Yep.
So is that the end?
No, we're still going. That was 300, but we're gonna do 500. So 200 more. more wood encrusted with not sure. More halosaurs as well. What is that?
What is this thing?
Is that looks like a jaw?
Yeah, it does.
What is
Check
that? Yeah, let's check that out.
Bones are good.
Bones are always good. Yeah, so the shrimp are probably also definitely feeding on the marine snow. They'll also eat
Definitely a job.
Yep.
a jaw of something. I was not wrong on the teeth. Yeah. Whoa. Is that a... That's a big jaw. And sharp teeth. Okay, so where's Diva?
Okay.
We have something
Okay, we're about to
interesting.
text you.
We were about to text you.
What animal do we think this is?
A shark.
Dolphin?
No, I don't know.
Teeth.
Obviously, don't listen to me. I think you should listen to the geophysicist.
Would you all want to park then?
Yeah, let's park.
So I think let's sit down.
Yep, we're doing it.
Some great imagery, really close up zooms and everything. And then, yeah, and then we try to collect.
Yeah, so just open the pool.
Maybe this is a mammal. Look at the vertebrae.
Yeah. Yeah. Those look like vertebra.
Oh yeah, that has also that for sure. Okay.
What?
Do you want to pass it on now?
Sure. Oh, go ahead.
Okay. So
So,
what we'd like to do
yes,
is
sit down, get some imagery.
get some, but like, so the first vertebra
Yep.
extends out all the way to here.
Yep.
So literally,
It's
just
that big.
zooming,
Yep.
panning,
Okay.
right?
Yep.
Tilting
Yep.
up the whole thing.
Can I sit
Yep.
here? 'Cause
Yeah,
there's
you can
like a
sit
little
wherever
bone.
you think works
There's
best.
also a bone right
There's a
there.
little bone right there, right?
Yeah.
You don't need to sit on anything or not.
Yeah, so it's
Might be a little far, but
Also keep in mind.
like
we're
it's
talking
over.
about
Don't worry about that one.
don't worry about that.
Just sit wherever you need to get
Yeah.
the best imagery.
Because that there's stuff over here, Diva. But
Like that, facing that. That would be pulling my umbilical towards the boat, which would make it very backwards. So I'm gonna scan this.
So then
Yep.
once we've like literally
Panned and
zoomed
done
until
mowed the lawn.
mowed the
Yep.
lawn kind of sitting here.
Yep.
And then we get if there's any amazing animals and like
Yep.
really put close zooms of those. And we think there are ostodax on these bones, which are the bone-eating zombie worms.
what?
So when we look, but it certainly looks like there's stuff on the bones.
Yeah,
So
sure.
then we're gonna collect.
Do we want to collect the jaw?
So Anna was saying the jaw.
I would say the jaw, but
and then
I don't know.
maybe- Where's the second laser?
It's
We're just, it's because of the
Does this look
push
like
board.
a
Ah,
Yeah.
yeah. There we go.
There it is.
So is that the ostodax?
Yeah.
Zombie
so
worms?
Those are all mucus tubes of ostodax.
Yeah.
Wow.
Yeah.
Definitely getting the job.
So we're gonna collect a few pieces. Oh my god, how exciting!
Mhm. That is very cool.
This is pretty rare.
This
Yeah.
will be the first ostodax ever collected
What?
of this number in the Caribbean, and the first in the Southern Caribbean.
I was gonna say, what about the Atlantic Inland?
So Caribbean, they have been
Sure.
from like mid-Atlantic.
Yeah, but this is pretty dense.
No, this is super dense. Yeah. So we're gonna just try to collect
Yep.
several, definitely the jaw. And,
Okay.
And then
Yeah,
maybe
I'm
a few
just saying
vertebrae.
vertebra.
Yeah.
And that
But-
looks like a rib to me.
But if we
Mhm.
can,
This is a rib.
so yeah, but we really want to mow the lawn so that
Yep.
we get like
We
a
gotta take a
full
picture.
Yep.
systematic view
Yep.
and like some
Do you want to fly over
really
it or
close-up zooms of
these,
when
all
we're
these
done.
little?
Like
yeah.
these are for sure all ostodax. So we want
Yep.
beautiful in situ imagery of these because these
Okay.
will for sure be new species and they will for sure, this will for sure be a paper that we work on about this. And you can actually see all the reducing sediment below it
Yep.
as well.
Yep,
This is
these
amazing,
are
guys.
bacterial mats, right? The little, yeah.
What?
Okay. What's that? Dolphin?
What do dolphin teeth look like?
I mean, the best chance of ID in that one would be
Oh,
Yeah,
yeah.
so absolutely, they're taking it.
It's good stuff.
like little amphipods, little snails. So we might also be a little polychaete underneath. So we may, in addition to collecting bones, we may also want to do a quick slurp.
What's that? Can we zoom in on that little?
Things we weren't expecting today.
Actually, Max.
Are you? Are you? Sorry.
Let the excitement
Well, which I could
go.
do. I've got
I've
got a big
I got something.
I'm watching the phone, but
but
I'm
if we're
just
gonna
thinking
grab
what
these,
happens.
I
Yep.
This
No,
is
you're
really-
right.
I will,
That's
yeah.
you're smart. Never mind. I just want to like
Yeah, looks like
little amphipods or
scale worm
shrimp.
thing.
Oh, look at the little. Yeah, they're swimming around in there. There's
Here's
one
some
right
closer
there.
too, so-
Yeah.
So
Oh yeah,
there's
yeah.
polychaetes under there.
And this little. Wow. There's a whole city going on in here. Yeah, we don't think it's. I don't listen to anything I say. I'm a geophysicist. It's not a shark.
So, sorry, I missed this. I'm starting to do that. Did we get really good?
We're going to go pan. We're going to do like
Okay.
back and
Yes.
forth over the whole thing.
Yeah.
We were just right now still overwhelmed by it.
You want to start? doing
Yeah,
that, or-
let's do the just
Okay.
sort of mowing the lawn over the
But like
but
stopping. and
zoom in.
everything.
Yeah, zoomed in and going up, over, down, over. So we get a way to stitch it all together, maybe in close-ups.
Certain fish, but we're not sure what kind of fish.
Yeah, those are vertebrae. Wild. Yeah. Look at them. Cool.
Yeah. So we'll take a slurp up. Maybe a little slower. Whoever's going to panic. You can't tell.
Max,
Mike, or is that you?
that's me, but at max zoom, that's kind of the slowest. I could zoom out and it would be slower.
Maybe a little, yeah.
You zoom out?
That's
Okay.
That's good. Yeah.
Okay. I got a sunny here.
It's
Sunny, you have a Sunny behind you.
So this is a very
We just found something very cool.
find here.
Mhm.
So we're just kind of trying to do video of everything like
We're doing like
back and
mowing the
forth,
lawn.
back and forth,
Okay.
back and
Yeah.
forth. And the scientists are mostly just trying not to hyperventilate.
Yep.
You might have to get
Keep
them-
calm.
Trying not to get them some paper bags.
Tell them to slow down.
Well,
Calm
obviously-
down.
Obviously,
Well, can we zoom into this too?
Yep.
Sorry.
this right here.
Yeah.
That's a tube worm,
Oh yeah.
isn't it? Yeah, there's a little.
Obviously, be really
Well, there's
careful
something-
of the viz. I got the current's blowing towards you,
Mm-hmm.
so it shouldn't blow into it. And I parked a little bit far back just so we don't disturb.
Oh, yeah.
There's like, because they might want to
'Cause
take
there's-
that or take that
yeah.
or take that. So I'd be careful if you move forward. This whole area is like a
Uh,
fall, right?
Yeah,
Okay. There's one under here, but they said don't worry about that.
yeah. Maybe that's from the tail or something.
There's some cool teeth on this thing.
Yeah.
What do they recommend it? Or what?
Wait, I don't think we know.
I don't know yet.
I wanted it to be a shark, but it is not a shark. The teeth are very cool.
Let me know when the.
We're good to move on. Yeah. Thank you. And then we'll just keep going. We kind of did that one, and then we started going down, and now we're gonna go do the same thing, but over the whole area. probably stop there. Go now slowly, but zoomed in a little bit. You want this zoomed in, right, Anna?
Yes.
Okay.
definitely.
Yeah, so a little more zoomed in if we can. Yeah. And now just sort of slowly panning up. And we'll keep doing that so we get the whole area, because where things end up may help us determine what it is. Yeah, and there's another one. That's it. Huh. That's probably it, I think. Hmm. Well.
Suppose we go moment, please. Yeah.
Well, yeah, there's like whatever, these little things hanging on the. What did Diva call those worms?
Osedax worms. Osedax.
Osedax?
O-S-E-D-A-X. Osedax.
Osedax. And there's some zombie something?
I think, yeah, but I don't like this dirt.
Okay.
I don't like the zombies in the area.
All right.
Yeah, so it's just.
It does. Yep.
It gives the Dixie a bad rap. But yes, that's what they were.
Okay.
Also, that's great.
Cool. Who's up after me? Sorry.
Jamie, I think.
Where is she?
It's enough for another half an hour.
Half hour?
We got in early.
Oh,
It's on the half.
okay.
Again, if we can zoom in here.
Anna, what is the stuff that is coming up?
Jamie. It's a worm.
It's a what?
Yeah, but the only ones don't know.
No, it's fine. I didn't realize we got in early. So what are the little bugs on here?
Those look
amphipods?
Yeah, isopods.
Isopods?
Is it as close to the zoom as we can?
Yeah, we're maxed out. We can get closer, but we didn't want to until we saw everything where it is. Can we keep moving?
Just, yeah. Further.
Sorry, can we move to the south or south? That's good.
Sonny, you okay
Can
for
you
reset the
go
eyes?
here rather than here? Focus on the, yeah, this, yeah. Whoop. I know it's hard.
We are zoomed in.
Yeah. Now we, yeah. So, but we pushed the dive, in any case.
Oh, I thought that was out.
But, okay, so Rachel, you've been here since more than an hour, have you?
What?
Didn't you start at 9?
I started whenever we went in, whenever that was. It was early. We went in before 9, I think. Anybody know when we went in?
I don't know, I think it's something like 9. You don't wanna leave the hot seat, do you?
No, I mean,
Did they also find out? And then she said, okay, I'll finish it. No, it's just taking the samples. But she said, oh, yeah,
Oh, that's confusing, so.
yeah. So these are meant to be updated. And she said, Rachel, but these are the shifts that
yeah, what kind of
Okay,
bone is
alright,
this?
that's fine. So we'll follow the shifts and,
Yeah, I
Rachel
don't.
will do another half.
Yeah, I'm fine. I wasn't sure what the shifts were anymore.
I know.
Yeah,
that's
because
right.
it.
So
Cause,
this is the shift report.
Yes,
Yep.
Yeah, Rach,
all right.
together.
All right. What is that bone?
You know, the bone behind this bone.
The bone behind the
This
bone?
round one.
Yeah, I think that's another vertebrate, isn't it?
It's very large compared to the other ones.
Ooh.
And,
Should we keep going
yeah, just
on
make
our
sure we
grid?
follow the whole
Okay.
thing.
Yep.
Just trying to find bones here.
Yeah. Cool. Is that a tube worm or a bone? It's. Yeah, it's nothing.
A marker? I was gonna. Oh, wait, a waypoint you mean? Definitely, yes, definitely. Yeah.
Okay.
Yeah, bones. We don't know what kind of bones they are yet.
Yeah. Cool. Hmm. So if you are just joining, we have come upon, oh, cool. Now we're at the jaw. Okay.
So
Let's take some time here and zoom in and maybe,
yeah, it will be the second pass, I think. You know, we'll go up.
yeah. So what, all right, I'm getting ahead of myself here. Okay. So we're basically zooming in and looking at this fall of some kind of animal that is now covered in these worms that are apparently rare to see, encrusting bones like this, and trying to check out not just the position of the bones, but what is living on and between them in these sediments before we collect a sample. You okay to keep moving, or?
Yeah.
Yep, keep heading up. It's some kind of mammal. There's definitely cartilage and bones that suggest, oh, what is that? I mean, that's the biggest piece we've seen so far, right? Huh. Okay. Alright. I think that's it. And then we can go that way a little bit. Did you get an estimate of how long it was? Like the extent of the bones?
I guess
We'll get that, yeah. All right, so we're gonna zoom in here a little bit. Are those are the teeth, and we're getting into the jaw. Does not appear to be swim fishing. It's more like a small, like an animal that fell to the bottom and was picked over by lots of critters. And now what's left behind is what we're primarily interested in, I think, is the jaw, which would be helpful in identifying exactly what it was. Cool. Okay, we can probably keep going. Yeah, because those are teeth. Are these also teeth right here?
That's what it looked like.
Yeah, so there's teeth. We're zoomed in all the way, right?
Yeah.
Yeah. So they're over here, down here. Yeah, I guess we can keep moving. We okay to keep moving, or? Okay. Head down. Or whichever direction we're going, I don't remember. Down. Stop there.
Stop.
Okay.
Stop.
Thank you. Huh. Yeah. I have no idea. Ready to keep going? This way? Hmm.
So there's all this other polycade tubes here.
Yeah. Huh. Yeah, so this is a fish fall, most likely a fish that, as it died, became a place for other life. And what we are trying to do is catalog and sort of take a census of all the small animals that are benefiting from these bones and the jaw, and then also trying to identify what it is. Oh, look at that guy swimming. That's cool. Huh. Earlier in the cruise, we saw a ray fall, which was an incredible find. This is, a dead large manta ray, I believe, that died, and we found crabs feasting on the soft tissue. And that was probably one of the best examples of such a fall. And now we've been lucky enough to find another one.
Do you even have swordfishes in this part of the world?
What's that?
I don't have any from here. Sorry, I thought Brian was such a saint, but Brian.
Oh. Are we okay to keep the sweep going?
Keeps with-
Yep, okay. Okay, we can probably go over. Huh. Oh, there's, what is that? Wow, okay. Wow.
Let's stop there. Oh, no, yeah.
Well, we weren't there yet.
Oh, I see.
Yeah.
I thought you were already.
No,
This-
we're going down still. Okay, that's good. All right.
Stopped before as well, not just for the thing, just to really, you know, things around. Yeah. Great.
Hmm. Hmm.
It's good, Sunny, thanks.
All right. Huh.
Okay, stop here. So we're trying to identify what's going on around it in addition on it, yeah. So the sediment, discoloration, all that stuff. That's good, right? What's going on here?
Hmm. ready to do the last swipe, I think.
To go down.
One more? Okay.
Oh, okay, look at those.
Yep.
Worms.
Which one?
You know the worms, there are all those squiggly worms here on the
That-
right
Yeah.
side?
Okay to keep going? Or- So what exactly we want to sample?
So first, it would be good to sort of zoom out a bit and get an overview with the lasers so we get an idea
Yep.
an estimate how big it is. So let's do that first, where we have an estimate. We do want to get the bones.
Well, not all of them,
No,
obviously.
not all of them. But we also want to get suctions around the bones. So my thought, and I'm looking to the pilots now, is if we pick up the bones, we're going to disturb everything around them, right? So we probably want to do a little suction first.
Some pieces, and I'm not sure, but the large pieces, with a little suction, you can use the suction too for the damage.
So the bases. Yeah, but no matter what, once we start moving in, there's all these things that are under there. So we probably want to suction around the bones first to get the stuff, not the bones, the stuff that's
Okay.
underneath the bone or on the bone. And then, once we've done that, then we can lift. We'll see how to get the bones best.
Yeah, because I don't think fits on the scoop.
Oh, no, definitely.
We're not using the scoop.
No,
No, no, that just makes a
No.
giant mess. And
No, not the scoop. Okay, so if we could zoom in on the big one.
you mean the jaw? And the-
Yeah, around
and
that
the one that
area,
looks like a fin?
This whole area
Yeah.
here. So if you see there's also, so if we could just suction like gently around and under the bones before we lift them. And that will have to go in jar 2, please, for sure, because jar 1 has a very large mesh.
Okay, so we are suctioning into number 2. But that's what's about to happen.
Oh, Shirley wants a Niskin.
All right, first we'll get a Niskin.
Shirley, we might as well, I don't know why.
Should probably have Diva here.
Okay, I'll be right back.
Cool. Niskin number 1. What number is that?
It's 580.
580?
Take cables on.
It's there. It's our way of like to work. Philosophers. They steer it on my side and it fit perfectly.
Cute.
Techno-organic
So the objective here is to get not just obviously the bones that are left behind, but to first get an overview of where everything is at this stage prior to doing any sampling. But we're also very much interested in the associates that are colonizing and benefiting from this fish fall before we get to the bones.
Very gentle.
Yeah.
That means more on the commercial switch.
So,
We need to do a test somewhere.
Okay.
You tell me how much. Let's get to this little piece here.
okay, no, but we're not.
No
starting with
bones.
the
No. No
We're
bones.
not.
No,
So,
I know, but I want to show the switch first for you to decide.
okay, hold on. Let me tell you what I want, and then you can tell me what the section is. So, if we could zoom in. a bit.
Over there.
So, if we, you know, as I don't know if you guys noticed, when there's a whole bunch of stuff that's under the bones, near the bones. These are animals that are, but they're not attached to the bone. So we want a sample of that, and that should be very gentle suction.
If a bone comes with the suction,
If it comes, it comes. Hope, you know, let's minimize that possibility.
Try not to,
but
but-
if it does, it does.
So it's
because
just this sediment around the bones.
that's
water,
it,
right?
and all the
exactly.
little animals, right?
Yeah.
Now the thing about if a bone comes, I'm worried about tearing the mesh, right?
Yeah, because we have fine mesh in number 2, which is what we're going to be using,
Yeah.
or finer.
So that's one section. Then we'll change the jars, and then we'll try to get another section a little bit further away, still close to the bones, but a little bit. So what we're trying to see is animals that are like really attracted to the bone, but then there may be ones that get close to the bone but not that close. Exactly. So we want one just within the bones, and then we want another one maybe around there. Okay?
Okay.
So that's the, and then we'll pick up the bones.
just to confirm on that, you don't want the sediment.
Yeah, oh,
coming
sorry.
off the
No.
bone. Just
All right.
Around the bone.
All right, so we'll be here for a while, and Jamie is taking over.
some of the sediment from the bone.
Okay, let's try to make, so that probably
I
should
saw
have
that.
gentle section in the rest of that. Because it will come up with the bone and you pick up the bones, but let's just, as, you know,
You just tell me how deep you want. I'm going to
Oh.
start on the surface.
I don't want you to go deep. I want you to surface. I only
Tell me if you want.
Good morning, everyone. My name is
I only
Jamie.
want
I am a
surface, Matthew. I don't want
grad
you
student at
to
the University of the West Indies.
So also, I think it's important.
We are not 100% sure on which animal carcass this may be,
Maybe we should start
so we're
away
about to
from
collect
it, shall
samples.
we? Because then
So
it's
bear with us.
easier.
It's going to be a
So
process.
maybe, like, do a section around here first.
This side here is easier.
I know, but this is, you see how the sediment is discolored? So that would be preferable.
More or less.
Sorry. I know we're not making your life
More
easier.
or less here.
Like here, however, so what I would like, if possible, to do is sort of take the section and do this. Right? Not,
Exactly.
yeah, so try to get what's at the surface, not necessarily go into the sediment. As much as you can,
Not
just
with
get
the-
as much surface area as you can.
What is distance? Start a little bit,
Is it, sorry, I don't know, I've never used a second something.
Okay.
Is
So-
it easier to do it vertically? Can you fold it vertically so you can do it like a hoover? As opposed to like this? What about if it's
Yeah.
like, you know what I mean? Fold
Yeah.
it like this and
Yeah.
then go like this? Yeah, yeah, that style.
Okay.
This is going into two.
Should be.
Should be able to come down with the shoulder right
And
a little
pull
bit.
it.
Alright.
Pull it far.
Because it's far from the bones, and the other one's gonna
I
be
think you
on
have
the
enough.
bone and
We
just
have
going
enough
away
stuff.
from the
Let's
bone.
Yeah.
try it.
Oh,
No, well, say with your left laser, that as the furthest you're gonna go to left.
Yeah.
You want him to start it here and come towards.
So because we want to have a
the-
better understanding,
Can you
Oh,
go
one
this
second.
way? You think? Start here and then go that way?
Okay.
Is that possible?
Yeah.
Gentle section, yeah. We're just trying to get what's at the surface, but they're not gonna be big.
Can you turn on the lower right?
yeah, so because we want a better understanding of the colonization process of what's consuming the bones, what's around the bones, we're going to take a various set of samples near the bones, a little bit further away, a bone itself.
Go
Yeah.
away from the RV a little bit.
Yeah.
Yeah.
Because this is
Keep
pretty
going
rare
away.
to see, so
Yeah.
we want to make sure
Yeah,
that
a bit more.
Go away.
we do the best that we can.
More. 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 Okay.
Yeah.
So if you go there, come this way.
Yeah.
So come down easy right there. Yeah. Down. Down. Yeah. Good. Right.
Yeah, perfect.
So now
And
just
sweep
come
it.
to the right. That height is good.
Yeah, looks perfect. Perfect speed, yeah. And more towards
Come,
you.
come.
Yeah.
This way. Towards. To the right and towards the RV. Yeah.
Yeah. Maybe a little. Yeah. Again, we don't want to go deep. Just want the surface. So just
He hasn't touched the
no, I
seabed
know.
yet, though.
Yeah, yeah. Just sweeping. That's perfect. Like that looks exactly what we need. Yeah, perfect.
Yeah,
Yeah,
yeah.
there's some
do.
stuff.
It's basically what we're getting there is some sand, like when you see the little
Yeah.
vortex, but
Yeah,
most of the
little
time.
sediment. Yeah.
All right.
Okay.
Go that way now, up a little bit. Yeah.
Easy way to sweep it out.
It's probably easier if it's, well, not easier, better for the sample if it's a continuous move. But I think you have, if you could,
You want him to start again, more or less here?
yeah, not too close to the bone. Not so, I need you no farther than here.
More or less where I am now?
Yeah. And then come this way.
I'm closer to the very.
Just ask, what about all the cores that we have? Do we have cores? Can you sacrifice 3 cores?
Okay.
Well, the sediment is not gonna stay in that weight. It's supposed to form.
Sediment's not gonna.
Okay. So let's,
Still waiting on. Still waiting on.
yeah, there's thumbs that are coming up. Okay,
Oh shoot.
so I think that's good, Mike.
She's responding.
Or that the distance.
Oh.
Sure.
Okay.
All right, so now, we need to rinse this slurry, right? This is where we use dry one, because we need whatever is in there to come off. Like, we don't want to start cross-contaminating.
Okay, what you can like run very low flow of heat on the
Sure. Yeah.
And
Yeah,
then
just clear this, we just need to clear the whole hole, so whatever
exactly
will go.
let it
Okay.
run a little bit of the water.
Okay, yeah. Okay. All right, there's another call. Okay, so, are you clearing it now?
Yeah.
Okay. So, before I get all that mess there. So I think we should try switching jars, right? Going to jar three.
You want 3?
Yes, please. And so this is where you're gonna do the same, but around the bones.
Okay.
Close to the bones. Just trying to get it back. So let's try that.
So just on top of the bones, we're not trying
Not
to
on
move
top.
the bones,
Not
right?
on top, because
Okay.
the bone's gonna come around it. So you know we saw all those things
Yeah.
underneath it crawling? Yeah, that's what.
Moment, you walk
Yeah.
the same way or you don't.
Well, you're gonna have to go sideways, because if you go top, you're gonna get the bone. Yeah, so
No,
you,
no, because it can come on the top, but just around, but you want what is underneath,
exactly.
nothing in the middle and.
Yes, yes. We want what's underneath. Yes.
Okay, so.
Yeah.
The other side will be more difficult for Anna. I'm gonna start, I'm gonna do the easy part
Yeah, okay.
and then.
Do the easy part, and then we'll take it from there.
Yeah, then we decide to do the
Yeah,
other side.
Yeah, let's see how the easy part goes.
I'm gonna start more or less here is good for you. And tell me how close I can go to the bones.
Oh, you need to get underneath the bone, so you have to get pretty close. But
Instead of going that far, Mike, you wanna just try underneath the jaw
Yeah.
bone right there first? So it's a little closer there. By the jaw there.
I just don't want to make a mess in between myself and the target.
Okay. Okay, go for it.
How much switch on there?
A little bit.
okay.
Can I touch the soil?
Ah, sure, of course. Yeah, because the animals are on the. So we will bring the bone, so we'll get whatever's attached. So definitely, we're so close to.
Yeah, that's fine.
This is in 3, right?
This is in
Yes.
this. So
Two.
this is on the bone. Okay, that's probably enough from that side. If we can come around. more.
Near.
Here. Like where, yeah, the jaw. Yeah. Let me get the pointer.
Any guesses as to what you think it is?
Oops.
We have
On
no
this
idea.
side.
We're thinking maybe swordfish.
Yeah, around here. And then
Soft
around
shot T10 now?
Yeah, we were looking. Trying to look and compare them. Kind of. I'll figure it out.
Are you as soft as you were with the other one in terms of the suction? Or is
Uh,
that stronger suction?
Probably a little stronger. I'll reduce it a little bit.
Let's soften it. I think it draws softness. Yeah, it's a little too soft. So
Yeah, I think it's because he's touching. That's why it's looking like it's.
Yeah, okay, there we go. Yeah. Okay, so I'm gonna make it slowly coming out. There.
From the top down, then
Here, in here, yeah, wherever, however you can get it, just this part, this stuff under here.
also, I mean, why not? I want to see.
He's watching. What's going on there? I love that spot.
And then you can go up and down as well. Well, just if you touch the bone of it, just to get this stuff. Alright, so maybe we can move over again.
Oh, no, oh, buddy, the one with the teeth.
Oh, around the teeth here, right? Yeah. Are you making contact?
Chinese. Go ahead. No, I'm listening to a message.
Oh, I see.
Wow, those worms are really attached.
Oh yeah, they are. They're in there. Their roots
Yeah.
are in there.
It's
Do you
incredible.
wanna kind of scrape the bone to?
No, no, because
No?
we're not gonna. Oh,
Oh, let's
no,
drop him.
Let go.
Yeah. Yeah,
You
yeah.
want
I know.
We'll get him,
Alright,
we'll
so
get him
then
after.
Let's move over to the bigger pieces there and do the same. Yeah, like here and then there. Yeah, yeah.
Before the sargassum, below.
Yeah, yeah, below the sargassum. Yeah.
Okay.
If a small bone counts, that's okay. That's fine. I'm working with it. And then in there, get in there. Yeah, this part here. Looks a lot bigger than it is when you put the suction on top of it. Yeah, that's okay. And it looks a mess.
Yeah, when you put a suction, you're like, oh, it's
And then
not
up
as
to
giant.
this one. And then turn around there.
On top of those big ones.
Yeah, here. Oops, sorry. Sorry. Alright, okay, we don't want this aggressive. Okay, so.
Threw it out down.
Right now, once you turn off the pump.
Yeah.
It's stuck.
I think
Take
that's.
it to the right and bump it.
Yeah, there you go.
I think that's good. So let's move on to jar 4.
Want another kind of flashing.
Yes, definitely.
Procedure.
You're flashing. Yeah, definitely flashing both sides. Yeah. So because we need the cores for a different thing, we're gonna replace the cores with slurping. So you're gonna go in and now you're gonna fill that jar with sediment. That'll be jar 4.
Fun.
It's a little bit bigger for me.
You don't wanna go all the way to China?
I can go.
Shake-
The
shake
problem
your-
is that is one-way chicken.
Shake here, who's there?
Alrighty.
Give it a wiggle.
So, yeah, if it's easier, go back to this middle, right by where that shrimp is sitting, and just slurp until.
You want to slurp on the area where the shrimp sitting.
Well, that's because it's an area that it just happens to be where the shrimp is. It's the area where we did the surface before. So let's just go deeper in there.
Oh, okay. You want
So
to
do the same, like going back and forth, but we're now looking deeper into the sediment. Yeah, but the same area. That's what I said where the shrimp is. That's where we're working from.
If you want to go deeper, you want to go just with more suction like that, or you want to really go getting more?
Well, what is
of
gonna
the
happen
solid?
is you're gonna be going along and then, but if you come back, you're gonna be picking up the second layer anyway, so it's probably easier to do the way you did, but maybe go and then go a little deeper and then go a little deeper. As you're
Okay.
suctioning it, it's come up to the surface. So, yeah.
So the shrimp where he starts nearby the bone, where is the sargasso more or less?
Okay, I'm gonna show you.
Because for here, if you're open to do like that,
Yeah, that's fine, of course, we can do it, that makes sense.
sideways,
But
which is a
we're,
mile.
I need you to start maybe here and just go back and forth. So between this place and that, in between those bones.
What I'm gonna do, I'm gonna start showing you how they go like that. Very close.
Yeah, sure, we're gonna, just make sure you go over the same sediment over and over so that you get. So that's jar 4. It's going into jar 4, correct?
Yeah. A little higher up. Start from where the shrimp is.
I use the sargasso as a reference. You have suction?
Yeah.
So this doesn't have to be gentle.
No?
Yeah, this one we want to fill with sediment.
Okay.
Whoa.
Yeah, it's fine.
Okay.
Move it a little
Move
bit.
to the
Move
right.
it, yeah. See what's coming up now, it's sediment below, it's a different color. It's anoxic because of the bones.
Oh.
Trying to get some of that. Oh wow. Look what's under there.
Whoa.
Let's just keep sectioning deep
Yeah,
into
can we
that,
zoom?
please.
Here?
Yep. Just go into the sediment.
Yep. just
Stronger
push
section,
in.
maybe.
Full
Yeah.
suction.
Yeah.
You need to
Yeah.
do it.
Wow.
Okay.
How is it not
Very
full?
honest.
You'll see.
Oh, okay.
It's just little dust coming in right now.
True.
Now you'll start falling.
Whoa, there's.
There's all
There-
sorts of stuff down there. Just
Yeah.
keep digging there, Mike. Alright. Okay, that's probably good there.
Yeah.
Wanna clean
Yeah,
the holes and change the jar?
Maybe change the jar and do it again. But we need to, you know, what do you call it? Flush first, please.
So we're flushing and then doing the same thing in another jar?
Further up.
Okay.
Yeah.
Shake.
Another jar. Yeah.
Will this be a separate
We're doing
sample
coring.
number?
We're doing
Oh,
coring
okay.
by section.
Coring by section.
Can I start the next line from left to right, more
Um,
or less here?
Don't get
A little bit further
a
up.
little bit of it more towards. Yep. That's
it.
good. Yeah. Oh, she wants this
And
pretty
the distance from the last to hole.
Okay.
So this is jar
Yeah.
5 now.
Jar 5.
And this will be a separate sample number?
Yeah.
Okay, cool.
Hold on. Okay, suction it up.
Same idea, yeah. And then go deep. Let's see what's going on.
Go deep right away.
Yeah, just
Okay.
go. We know what we're gonna see down there, so let's just get another sample.
Whoa. Whoa. What is that? Sorry.
Whoa. What are all these things?
What is it?
Other tuberums down there.
Yeah.
Yeah, that's good, Mike, I think.
Good enough.
I think that's good. Okay, please,
See if you see
flush,
something
please.
good
Flush, please.
Yeah.
Can I ask that we put the lasers on these things so that we can get an estimate of how big they were later? Yeah, that's great. Thank you. All right, let's do a third one in a different jar, please.
Yep. That's next.
Oh, fun is a work with the UXO.
What's that?
Work with the UXO.
Yeah.
And each is right around without to go the ball.
So let's maybe do the next one
More
there.
or less.
Yeah, more or less.
Is it?
Same length of that. Just go ahead. Once we're.
This is far from the ones as well.
Shake it.
They're all,
Yeah,
yeah,
it is.
I mean, at
Okay.
the end, I will ask you to take a picture, so,
Okay.
you know,
Alright.
capital.
No, I'm still on one.
Or sediment, yeah. The other ones were surface, obviously.
She wants one on the bones that we collect.
Preferred chlorine by stirp is the one that we want.
But then she also said, I want to show,
After we
I want to show what
collect
she meant by so do the normal close as well, okay?
I know one's chlorine, so no, that's not what Anna said.
Oh, sorry. No, that was from before, like before the survey.
This is fine, let's learn from
Me,
That's
ready now.
how she was.
Okay.
There's one second
Coming
first
over.
before, so it's fine.
Yeah.
Okay.
Okay.
How far ahead of this hole, and more or less here, or more close to the hole?
Smidgen over this side, this side. Yeah. Yeah, that's good. Perfect. Don't touch that bone.
Besides the bone, you mean?
Yeah, don't touch the bone though. Don't touch it. Alright,
Oh,
in you go.
touch.
It's fine. Yeah, just keep going deeper, don't worry about it. There's a lot. Gosh, who's gonna count? What poor soul is gonna sit through the sediment?
I think I know which person.
Okay, that's great. That's great, Mike. That's good.
Good.
Yeah. Thank you. All right.
Same thing, flush the hose.
Flush the hose for sure, because we're gonna need to do more sectioning later.
Yeah.
So now we need to think about picking up those bones. Can I get you first to zoom out just a bit so we have a picture of what we've done in case we destroy it when we try to pick up the bones with lasers and yeah. So do you want to get a photograph, please? Yep.
Huh?
All right, bones. So we don't want the bones in the slurp. We want them in a firebox.
But why we don't try the bigger ones? We are sure that don't go through this lump. Can you see how gravity?
I know you did, yes. So we will
What's the-
gonna need some of the big ones.
Just a gentle, that with the slurp and drop
Right.
in the other ones.
We can try that. Now I'll tell you which bones we want though. So we want the.
How high do you think gonna be those bones at this point? That's the problem.
Who knows? Don't know when you pick it.
That's why I'm afraid to use the jaw.
Right. So we want the jaw, for sure. We want this thing.
Uh-huh. The bigger one, that's fine.
We want
Yeah.
this, and we want. Well, let's start with those and we'll see how it goes. That should give you enough work for a little bit.
So we put these in the big bio box, I'm guessing.
Maybe five minutes. Start with
Yeah,
the
right.
jaw. Yeah, you could. I mean,
Think
the
the
general
big one?
slurp, you saw it
We're
started
putting one
coming.
for bones,
So
or
let's try
you
that.
think it could fit in?
And then
Could
carry
fit in
one
with
of
these
those.
holes?
Yeah. Let's try that.
Sonny, you want what's out first to be the closest as possible for me to cover so much ground.
I'm sorry, I'm totally overrating you.
No, this isn't learning experience for me, like how to manage this. Oh.
Plus that.
Yeah. Plus, yeah, when it needs to be done properly.
You right one?
Wide.
Yeah. Close there. Okay.
I feel like we should maybe also get one of the little vertebrates. Just
Yes.
because they have such high density.
We'll get the vertebrate. I don't know if these
Of the
things are
worms.
red, so I think we should get a red as well.
Yeah.
See where you can get the position, people don't come pointing.
Yeah, I'm off.
Okay, I think you got it. All right.
This is going in 1A. Sorry.
Oh.
No, it's not big. Not big photos. It's just good.
Yeah, yeah, yeah.
It's in.
Woo!
Perfect. Okay. You can breathe now. I can breathe too. Alright. Okay. So then the other one is bigger. It's okay.
Yeah, I was like, what's sticking out of this sediment? He's just buried. He's just buried under there.
Anyway, buddy, big one.
Yep.
Oh, but yeah, you wanna down on there, have all of it? Oh, for sure. Perfect. It looks like it. It's tail sticking out.
Yeah, he's just buried.
He can't see us, we can't see him.
Exactly. Exactly. Yep. It's heavier, eh?
Yeah.
Maybe the jaws for that one.
See.
Okay. What's that mean? Your bone's done for the shape. Oh, is that the skull?
Yeah, it looks like the skull.
Wow, like not drop it. Looks like a fish skull.
It looks a lot more. So
But last over. Okay. Palms away.
All right. Okay. It's going to do the same, right?
Yeah.
Good. Okay, let's see what else now. We're just going to make it a little more challenging for you because you seem to be doing well. So let's see how small a thing you can do. Make it a little hard.
That was a guy who you received this
yeah, he came out.
He came out of somewhere.
Look at him.
Aw,
Interesting.
curious.
See what's going on.
Yeah.
Which one are we going for?
Seems to be the underside of the jaw. Yeah, that looks like a jaw, doesn't it? The underside.
Where's on the other side? Oh, that
This
one.
thing.
A little more
Like
to the
there's
left.
more teeth on this one. On the right.
Yeah, there's one take there.
Yeah.
Oh, okay. Oh. Oh,
Whoa.
this is gonna be heavy. Yeah.
Yeah. Oh.
Try the thicker side. Now we come back a little bit.
More tooth.
Yeah.
Oh, yeah.
So it's
No. Oh. I think this one may need.
might need the jaws.
Yeah.
The flops. Oh.
Okay.
Okay, let's see. Hold on. Hold your breath.
Down a little bit. Okay, I'm gonna drop. It's
That's something else. Part of the bone.
Hey. Oh, are you on next?
No, no.
Oh. Okay.
Nope.
Just
It's
watching
Watching.
Mike sweat. Okay. All right, Mike. No, you just uncovered something. That's the problem. Where's that? Right. Okay, so under where you were, there's now some funky thing. See this thing here? That was, yeah, it was right under where you were, so you get underneath.
Talk about these
Talk
lines
about
straight up.
this. This. I don't know if it's one piece, but these are the teeth, so we want. Yeah. Oh, well, that's fine. And there's the other one. There's that little.
Let's keep doing it up like that. Then just turn off the switch over the phone inside the box.
Yeah, yeah.
And number one.
Oh,
Yeah.
Shit. There's some more stuff in it.
What's coming out?
This was that by what's this?
I was thinking of trying to find or go switches back.
Yeah. Drawer stuff in there, you need to come up a little bit with the elbow. And you press it.
Not the waypoint to perfect. Okay. So we'll just do that now. I need a few more things. So, we need some vertebrae, but I think those might actually go in a jar, right? Because they don't seem to be sharp. There's one there, there's one there, maybe something. Yeah, I'm using it. Let's have another pieces. Oh, there's one here. So there's one there, and then there's one right by where we. Just suction those, I think, easier. Just put them into the jar.
We can try suction because if I see that very tight.
Yeah.
I don't know if you saw that we
Yeah.
turn the suction over.
Sure, yeah.
It stays here.
Yeah.
Then I
But
just
they're
walk.
gonna be pretty smooth, so if you don't, if they can go, it doesn't matter what they go.
Yeah. I'm gonna come visually because what I'm afraid to consider a little more graduation.
Yeah.
That noise, because when somebody gets there.
Yeah, right.
I'm gonna
Okay.
come visually.
Okay. So,
Well, let's start with
One,
the
two.
order. What's the smallest?
there's one here,
Let's try
one here,
that.
one there.
And then you have a vertical.
So new jar, right? What jar is
Number
this
one.
one?
One.
Oh, you mean the new jar will be jar number 7. We have 2 jars left, 7 and 8.
Yeah,
Alright.
okay. But it's not going in the jar. You're gonna try to put them in the box? But let's
Okay.
have them on the new jar just in case.
Okay.
You know, the things go in.
Got it?
Still got the bones, huh?
Oh yeah. We're almost done. We'll just get those 3 things,
What is
another
it so
slurp.
important,
And we're good.
What?
What is so important?
Oh, that this worm that is growing, this stuff, it only grows in bones. And this is first records of Caribbean Atlantic cellular supplies. So you don't mind.
Okay, that's fine. Okay, big discovery.
Yeah.
Which one are you going for? Which one are you going for?
That one is below.
Okay.
It's there.
Alright, it's not coming down, I'll zoom in on. Sure. Should it go into the chart?
I think it's in use, so I'll stop
I
it.
think,
I think it's still in use, we might be able to shake it out. So, could we put it in the same buyer box?
Yes.
Yeah,
Everything
yeah.
goes in that buyer box.
Yeah.
Shake it, is it gonna fall to also? Because then I have more movement to be able to shake.
Okay. So you want it on that one?
Do you mind
Right.
if I use this one, because I'm gonna need to shake the hose?
Sure.
Yeah.
Circle under the boats.
Yeah. Then do the sediment, yeah. This one's not gonna happen.
Yeah.
You're pushing on it or? So. You got a lot of? Cement? So you don't wanna just suck it in section? We could try. Yep. I guess I'd come in. No? Yeah, Something big came in. Okay, so if that's the case, then let's just get just one of those section of that. No, that could be the center. Yeah, I'm just, yeah, you could see it then. Yeah,
Okay.
there it is. Okay.
Perfect.
The next, yes, and it yeah, you don't want that one, yeah, because that it doesn't matter really. Well, no, let's just here, this, can you reach that one? And yeah, this has the thing, you can reach this one. That's easy. No, this has a spine. I mean, oh, I don't know what you mean by flat. Back. Yep. What? Just trying to see if it's gonna land.
yeah,
Where's No, mine?
that's the old one.
No, that's the first one. Shake yours. No, it's
That's
still,
not it.
it's still to the end. Oh, it's Try up to there. shake it out under the porch.
Put it in BB2A if you can. Just with the others.
Yeah, that'd be good actually. Members of my boxes.
Yeah.
You could try to put it in this one.
Since you've been here.
I know. We let go of the section.
Yeah.
It's still stuck.
It's there. Look at that.
It's stuck inside.
Let's take a look at it.
Yeah,
Hmm.
we can see it.
Well, can we like knock it?
We took off Excalibur. We could have tried to poke it with
Yes,
Excalibur.
we can knock it against the box again. Yeah. Yeah.
It's proper stuff there.
Gosh, it's really stuck.
This
Oh, did it move a bit?
Like coming down.
Yeah, I think it's moving. Keep walking. Didn't it?
Yeah, I think. I think it's been
maybe
going. Yeah.
Just hallucinating.
let's take it out the take your arm out and let's do suction some sand and see if we can kind of slurp it down some sand. Easy.
Okay. Yeah.
Carefully pushing on the push.
Yeah. Oh, I think it.
Yeah,
It came out.
It came
Good
out.
job.
All right,
Good job.
perfect.
Woo.
Great. Okay, last sample here, and then we're gonna move on.
Yeah.
So next is we want to do another one of those sediment slurpy things right at the bones. Yeah.
Yeah.
But this, you need to be careful because it's gonna be bones, guys, and you're gonna get all stuck. So anywhere here or there, just below where the, under where the bones were, but in a place.
For the vertebra,
Yes,
we just suction
yes.
into
Oh,
the
no,
suction
you
sample.
want it-
There's
I-
one in there.
Are you giving that a separate sample
it's-
ID
you're only
to
doing
the
one.
bones that are in the
Right.
bio box?
Yes,
Yeah,
that one's
if you can do it,
587.
This is fine.
Yeah,
right?
let's make it
That's
good.
fine. But we want to be where, you know, the bones were
So look
sitting.
to the right, right on the very shrimp is there was a bone there that we took out.
Yeah, it doesn't have to be that big, right?
No,
So-
they just want you to suction right where you pick the bone up. You
Yeah,
don't have
and
to.
go into the cell.
Underneath where the shrimp is, we took up a bone from there as well.
And that's jar 8.
Yes, last job.
Well, we have one.
Oh yeah, we do have one. I mean, one's a little bit filled with things from filtering, but it is there.
And we can put like a
Yeah, something.
coral
For
sponge.
sure. For sure.
Okay, I'm gonna leave that over to you.
Good.
Thank you for the picture.
Under.
Jen is taking over.
Yeah.
Oh, sorry.
Hello. And can you go a little bit behind as well, I think? Yeah,
Further back?
like that way. Yeah.
Another place for me?
No,
Yeah, just right there.
Right where you were, just
Like
further
that.
away.
Sorry, can you point to what direction?
You were doing there, right here.
There before the right.
Yeah.
That's fine. All right.
We're good. The most all this process in the sand. Although we do,
Most I usually do, yeah.
well, then, over to you.
Yeah. I know. Got it. I'll give it a second. All right.
Okay.
I think we're good.
Perfect.
I also think we're ready to move on.
Thank you, Mike.
Thank you, Mike.
Woo!
That's funny.
Go for a rest time. We could take a picture again now that we have
How?
the,
Sure. You want lasers on?
yes.
Okay, I've seen one laser in the hole.
One of those in the hole.
All right, there we go. That should work.
I guess we're carrying out on a transect.
Mm-hmm.
So we've done 300 meters, right? We're almost at
Oh, this was during a transect.
400 meters. Sorry. Alright, carrying on with the plan.
Carry on with the transect.
So this was 400, right? Oh no, that's wrong. 400 is still quite a ways.
All right, Kyle.
Alright. Hold on.
We haven't even made it to waypoint 2.
Well, we're we're continuing. It's about to talk to Anna. I think you're going to continue the transect.
Okay, because we found this when we started transit.
Yeah.
Okay. Fair enough.
Samples, obviously, you can see all the samples for bones, Niskin, suction up like essentially segments to make bones,
See.
It's low.
It's a little hard.
Everything, all the suction jars, and very good. One,
Use it.
three. Yeah.
I
Still
never
really
use
wants
it,
it.
those suction jars.
There you are, for sediment.
For the first time I use it all.
What's that?
Hey, Mike.
Yeah.
How are you, Kyle?
Good, how are you?
Good thing I'm with you.
I don't know. Okay, again, any shots back?
Yeah, I think so. I'm just making sure.
Ryan, he fell asleep. He's probably going for lunch now. So we'll appear again afterwards,
Basically, we are at the beginning of the
Yeah, we haven't moved from the transit, I know, so I just wanted to make sure, because we've been here a while.
time-wise.
Yeah, time-wise. So let's make, what's the time? Oh, wow, it's lunch now.
Yep.
How long have we been here? We've been here long.
I didn't sit that.
Yes, I know.
We didn't sit here, Tim. 1 hour and a half. When you're extracting them, it's nice.
What's happening?
You'd be like,
Okay, you got your
every day,
shots
no?
there, Sam?
I guess we might as well, I mean, rather than wait, let's carry on to 400 and then
You
we'll
want to zoom in on the point for a picture.
which point? Oh, we have it.
Oh, you
We
did?
got it.
Did it look
We got the picture. Might as well carry on with transit. So, over to you, Gar.
Right? Okay. So we're just continuing on with the transect. Well, we just started. Is this like 300 or so? We haven't even started the transect yet, right?
We are on a transit,
We're on it. Yeah. Okay.
and we're just past 300 meters along the transit, so we can
Okay.
continue.
So it's a 500 meter one.
Yeah.
Okay. Okay. Yeah.
So we're headed to Station 3, right? From
Yes,
this
and I said,
Still following those.
You're going this way?
that's 500 is here. That's the 400.
Wait, are we going to 3 or what are those?
We are on the top of
Up 2, right?
300s here. We are in between.
We have another 200 to go. So we need lift off and
Okay.
Do you want to come back here or just keep going?
All right.
No, we want to carry on the line. That we have- yeah.
Yeah.
Bridge over here.
Aroge Bridge.
Please, may you follow us to the new waypoint?
Sure, no problem.
Thank you.
Okay, stop. We don't want to go to the transit. In the interest of time, we'll stick to the 300 meter. So Ryan, you may want to make a point that end of transect was at 300 meters, which we've already passed. We've just passed it. And so now we'll just head up to waypoint. Just turn it, go up the hill.
Okay.
Bridge, may you cancel that move? Plan change.
Hey.
Yes, first transit. And we were gonna go to 500, and when we passed 300, we saw the bomb. So we've done 300, we have the lights flashing.
Okay.
Sorry, are we transecting from 2 to 3 or are we?
We're not transecting, we're,
Okay, that's Russian.
no, we're not rushing it.
Not rushing to.
We're just heading to, just sampling. No, we're sampling, we're looking at things.
All right.
We're not in a rush.
Interview.
Are you in a rush? No.
All right, moving
Yeah,
on.
so just,
Okay.
yeah.
All right. Good morning, everyone.
There's some nice crinoids that we saw a lot of, so we may want to collect those as specimens.
Okay. Like
I
two.
think that's, one will do. I
One?
think that's what we see. They're everywhere.
Okay.
Yeah, let's get out of here.
You wait
And we
for
do
you.
that.
There you go, Liz.
And then sampling as we see things.
Mhm.
And if we start
No problem.
seeing it getting rocky, we
Yeah.
want to take the course.
Of course, around the rocks.
No. If it's getting rocky, we want to stop, go back, and take a
Take course before
Yeah,
Gotcha.
so we thought rather than take them right here, go a little bit closer to 3.
to the base. Mhm.
Yeah, but if we start getting,
Rocky, then we'll stop and take
okay,
course.
yeah.
Okay.
Yeah.
Alright, thanks. Okay, good morning, everyone. My name is Kyle Foster, and I'm a student at the University of the West Indies St. Augustine campus, which is in Trinidad and Tobago. And yeah, we are currently exploring the continental slope, which is within the UNESCO Man and the Biosphere Reserve, which is just northeast of Tobago. We would have had a very exciting sighting there just now of a fall. Basically, we think it might be a swordfish. Might have been a swordfish, that's just a guess, but we've collected some samples so that we can be able to confirm once we have on board. We would have seen lots of zombie worms or oceanographics, which is very cool, and this is something that's very rare to come across, so we would have gotten as much imagery as we could and as many samples as we could as well.
Look, do you see what we're on the INS?
Yeah, I'm gonna do a reset.
Okay, I'm on you guys.
Yeah,
now.
okay.
Yeah.
So we're currently about 755 meters below the surface of the ocean,
Okay.
and we're making our way towards the base
2 meters.
of the continental
Yeah,
slope.
there's DVL back in. Just give it a minute there.
And if you want to understand the size of the organisms or the environments that we're exploring, we've got 2 green dots on the screen, which are laser projections, and they're spaced 10 centimeters apart. So it helps with estimations. don't know what size were the crinoids. I just want to make sure I'm not gonna miss.
They're little.
Yeah.
Little.
I just want to make sure I don't miss any.
Yeah, they're very small, light.
Yeah,
Mhm.
Here's the sponge. Different person, yeah. Allergic to it.
All right, thanks. I'll keep my eyes open, but yeah.
They're very small, they're like this tall, and the crinoid is broad.
Okay.
Ultimate type.
So we've got a nice stalked glass sponge here. Oh, I think I
Yeah,
see.
right
Yeah,
beside
yeah,
it.
right there.
You
Yeah.
see it?
Uh huh. Oh, could we also
Maybe
please?
not this one because it's complicated. but
Not this
that's
one?
what was- Well, I don't know. You can, whatever you want. It's up to you. This one is-
Where are you going to put it? Because you sort of suction joints.
Right. Well, the crinoid that's like just to
Yeah.
the left of it.
That's the swimming one, right? That's the one that
Yeah.
swims?
The tiny one there. That's just above the lasers. So maybe it could go into like a coral quiver or something.
Is it the one that swims?
Is this like attached or is it swim?
It's a stalk.
A stalk,
It has
so
a
it's
stalk
attached.
and
Yeah.
then-
Alright.
But it won't.
Or you want something?
I don't think it will. If you want, we try. I don't
Mhm.
think.
Okay. So this one is fine to collect then.
Yeah.
Yeah.
I guess I should go and put.
Yeah.
We have no sections on this one.
Right. So we'll just pause and get some photos of it before we collect. And it really looks like the current's strong here because look how the sponge and the crinoid are like bending that way. And then after that, I think I see something interesting blue in the background there that we could have a look at.
Sure.
It's a blue water bottle.
Is it actually?
Yeah.
Oh.
Well,
This one?
maybe not blue water, but a water bottle.
No, there was like a
But
blue,
bottle.
interesting blue thing over there. Maybe it's some trash. Okay. So at the base of the crinoid, we have a brittle star. We're just getting some photos before we collect this as a sample. All good, Ryan? I mean, we should pause for like 10 seconds to get good video anyway. Good? Okay. And then,
So we do it for example.
yes, please.
The front of the one.
I can clip on the base, Kyle.
Yeah.
And remove. I'm gonna get with the plastic jar,
Mm-hmm.
like remove the whole thing.
Yeah. Yes, thank you. I like how you do it when you turn the lights off and it really gets it to focus properly, which is great. So we got this.
More artistic.
Yeah. Got this close up. You got Ryan? And then if we could get one with just the whole thing in there. Yeah. Got that one too? Alright. Yeah, thank you.
Comes in for this stuff.
And that's going in Coral Quiver 2. Which number sample would this be?
589.
589.
Okay.
Yeah. Oh, still on? Sticky.
Am I still at open edge or so? Still there, I think.
Just won't come off. Still on. Ah, perfect. Thanks, guys.
What's up with us? Fillet steaks. Is it? Fillet. Fillet McDonald. It's been good, man. It wasn't too bad. 2 knots or something dove in. Around 7. 7 PD.
Alright, Zach, there's something over there they want you to look at.
Yeah, just that blue thing. Yeah.
Oh, yeah, it's really good.
Yours.
Probably about that.
Ah, I see it now. Ryan.
There's a water over there.
One of those big water bottles.
Anthropogenic.
Very heavy now.
Heavy. Would you push cars or something?
Kinda in the jaws.
Kinda, yeah. We haven't even done the push course yet.
Oh God.
We intend to.
There are a couple of bones and stuff in so we're not as light as
My copy.
Thanks, Mike. Appreciate it. Good job.
yeah.
Good?
Yeah.
Alright, so y'all wanna go over that blue thing or?
Yeah. Well, yeah, we'll just like a quick look and take some pictures so we can see what species are interacting with it. But yeah, but nothing too serious. Is that a Venus on it? Yeah. They wanna.
Haven't heard from them in a long time.
Along with some crinoids and tube worms.
Oh, finally, it's up there.
Oh, wow.
Maybe we could have just taken taken that.
Let's go for it.
We could have, yeah.
Straight to the bottom.
Yeah.
Barnacle too.
Yeah. Interesting.
Okay, go over there.
I mean, I think we got some photos there just now, right? Yeah, so I don't think we need to.
Okay. Did you just get in here, Kyle?
Yeah, like 11:30. Mm-hmm.
Roger.
Okay, yeah, so we're just gonna continue to waypoint 3. And we also want to do some push course. So before it gets like too rocky, if we notice that it starts to get rocky, we'll take some course before. Okay, so we're continuing with our exploration, and of course, while we are within the UNESCO Man and the Biosphere Reserve, we are at depths that have not been visually observed before. So this area is pretty much unexplored.
Are you saying man and the biosphere?
Man and the Biosphere. Yeah.
And so is there like multiples of those?
Good question. Ryan, would you know? Are there multiple Man and the Biospheres?
Nothing Trinidad.
Not in Trinidad.
But it's like a worldwide thing.
Ah.
So what's the difference between that and a regular UNESCO World Heritage Site?
Good question.
I believe it's more of an area where you have a kind of coexistence.
Mm-hmm.
Yeah.
So lots of use, but still trying to be used in a sustainable, environmental friendly way.
A lot of interaction kind of.
Okay. Okay. Makes sense. So, in terms of like interactions, I mean like fishing and stuff like that around here, yeah. So we're seeing lots of stalked crinoids around here as well. We would have collected a sample just now. There goes a Venus flytrap anemone. And some fish, looks like.
There's a lot of cool fish.
Yeah.
Out there too, they're out there.
Oh yeah, I saw we were in the wet lab when that came on. And then we had the fall. Basically, we assumed that it might be like a swordfish.
Is that what the consensus is?
Yeah, I
Swordfish?
think so. But I mean, we collected samples, so we'll be able to confirm once we have in hand.
If they're moving too many, next time if you want, you can move them kind of on the other side of the line a little bit, you know what I mean?
So you're on the north of the line.
To the south of the line, that way.
This way?
Yeah, basically you're trying to always 90 it out from your launch point, unless there's some reason not to. You know what I mean?
To go and slowly start on your own.
Oh yeah, it's not an emergency, it's just next time you move it, you know? Just gives you that much more wiggle room to gotta do something with that. All those bones were exactly at that weight.
Bridge RB.
Yeah, go ahead.
Hi, Rafa. Updated the waypoint. We're going to move the vessel back to the south anyway, slowly.
Yeah, go ahead.
Thank you.
Another Venus. It's attached to something.
No, there's something under the seabed there.
Mm-hmm.
Like a rope or a cable or something.
Yeah, something like a rope, maybe. Or a branch.
Okay, yeah.
Oh, it's like yeah, really covered by the sediment.
A lot of wood around here, huh?
Hmm. But it doesn't seem to have had like gone under the different stages of woodfall that you would expect. I don't know.
Never so much.
Beautiful colors. Is this a rock hair? Some crinoids on it as well.
Sonar yet.
This is another Venus. Fly
On
chop
wood.
anemone.
Another one on wood.
Again on wood. That's yeah. It's a nice jelly there. Yeah, interesting. That's a pretty fat. Pretty fat as well.
Right there.
Nice.
Diameter of the cliff or whatever. Or more.
Can we do a little zoom on this here, please? This clear looking thing. Yeah. I think so, yeah. Okay. Thank you.
Which are, it looks white on the top, rough, you know, just slightly on the
Wait, what's that? It says on it.
on top.
Oh, tripod fish over there. Yeah. So this is a tripod fish. Typically they tend to face into the currents.
But we know what the fish is, right? Maybe
Huh?
that's-
Been seeing a
Fish we
Yeah,
know.
it's an inside joke and fire.
been seeing a lot of branches around here as well. tree branches. So I wonder if we might see like a larger woodfall or. Looks to be a rat tail. Yep, rat tail. And there's a little brittle star and a shrimp as well. There's quite a large shrimp there.
Even the big ones want to use fall like this or something. They're going like a kilometer.
Bridge RV, Tom, that's the white one.
Okay, wait.
More branches.
They're going like Joe's.
Yeah. Yeah, they're in there. Sargassum or rock?
Maybe a mix. There's another branch. There's a branch with sargassum.
Yeah, lots of wood around here.
Fishing net.
That too. Interesting. So you've got sargassum, a branch, and fishing net there.
Top.
Mhm.
It's brown and sticky.
What's brown and sticky? I don't know what.
Stick.
Oh, it's such a bad joke. Right? So many little rat tails around here as well. And more wood. So I don't know, Ryan, why would you say that we're seeing so many branches around here compared to like other sites? Mm-hmm.
to drain into this area, so it's possible that it's just bringing
Current's
some-
just carrying them from out here. Is it more fishing gear again too? Sargassum. Sargassum and branches. Yeah. So Ryan, who's from Tobago and is currently logging right now, says that there are a lot of like rivers within among like the northern range, so possibly why we could be seeing more branches than usual. The area is just probably a catchment area, or
the main ridge.
I'm sure that's right.
is that?
Kind of.
Huh?
The main
Yeah,
ridge.
that's the
Main
Did
ridge?
I say northern
Is that
ridge?
coral?
I think so.
It's the
Main range? Main ridge, not northern range. Sorry.
I'm not sure that's wood.
That's a rock, right? We think that's rock.
But then what's coming out of it
I
there?
think maybe it's coral.
Old coral?
Might be a coral.
Coral rubble.
Ah.
Interesting.
Hmm.
That would be a good sign for this cliff we're going to.
So yeah. So we're noticing more and more rocks then, and old coral rather than branches.
I mean, I'm no scientist. I don't lie to you. It's weird how it's attached to that. rock.
Yeah, makes sense. Yeah, it makes sense to be an old coral.
Supposed to look like it had some sort of tube-worm shell on it.
Yeah. A little squat lobster here. Next to a stalked glass sponge. What is that? An anemone?
I think that's like the one we saw before, but it's face mask. It's in a bottle.
Oh, wow. And the brown stuff that's behind.
I think that's the bottle, right?
A plastic bottle.
That's a
Plastic bottle. Yeah, it's a
Oh.
plastic bottle. That's
Okay.
crashed.
Makes sense, yeah.
So,
So we're noting quite a bit of trash around here. So if you're just tuning in, we are exploring the UNESCO Man and the Biosphere site or reserve that's northeast of Tobago, and we are at depths of 732 meters below the surface of the ocean. And while it's a well-known site, these depths have not been explored before. So we're here to see what we find. So I think at the very beginning of this, we came across a natural fall, organic fall. So it's just like bones, and we're trying to figure out what species it was. But it had some bone-eating worms growing out of them, or zombie worms. And that was pretty cool. So we've collected some samples. The worms are possibly new to science, so we'll see. That can only be confirmed once we have them with us.
What are they called? Zombie
You know, that's a good question. It's like, I feel like zombies usually go for like brains rather than bones, right? But yeah, we just call them bone-eating worms or zombie worms. It's a nice one. A halosaur. The way that its tail moves is just very like mesmerizing and hypnotic.
Pete. Pete, come Pete.
Oh, nice.
Good chocolate. I haven't tried it though.
Was it the chocolate?
So it look like.
Okay, some more.
And it's ice cream, right?
That's the night.
Yeah.
More
Just have
coral
to
rubble.
plan my desserts.
More coral rubble, I guess then.
It's marvelous, these.
So yeah, so we're currently at heading towards the base of this continental slope, which might be rocky at the top, we anticipate, and we've got some certain clues that that is the case, because we're seeing bits of, as the pilot, Zach, has pointed out, coral rubble. We thought there were branches, but possibly like old coral now. That looks like a leaf. Oh. I feel like we look at the same things at the same time because I'm like, wonder what that is, and then you just automatically zoom into it anyway. Okay, yeah. Some more coral rubble.
Coral on top.
Yeah. Are those?
Very
Are
old.
those shrimp or?
There's some arms sticking out.
Oh yeah.
That's one of those crinoids, huh?
Crinoid, yeah, we just collected one of them.
Yeah.
They did.
and then the brittle star is just under trying to wait for help, maybe. It's the base of a sponge.
Some kind of shell.
Hmm.
Not zoom. Sit down if you want.
You want to get a closer look at that?
Sure.
Yeah.
For Charlie. I'm closing it.
Wasn't it?
Well, I was wondering if it. Zach says shell. I was wondering if it was the base of a
No, it's a shell.
shell. Hmm.
Keep going. Report.
Yeah, keep going. Thanks.
For Charlie. You
Anyway, I'll stop this.
That's pilots here. And they're going. What's going on, Zach?
What's up, bud?
Partying. What you doing? Shrimp party. Huge. Yeah.
Looks like a massive one. That looks like wood. Got some holes around here. A little fish peeking out. It's retreating.
Brighter, brighter.
Oh. I wonder what kind of fish it is. Yeah, it looks like more trash. Wire, yeah.
You know, move to the Blazing Cliff. Copy. Oh, you speed the boat up a little bit, or you got this?
I think we can speed up a little bit, right, Anna? Yeah.
Port Charlie.
Go ahead, Harvey.
That's all. What's our current speed?
I've just reduced us down to 0.2. We were moving at 0.4.
Can we go up to 0.6? We're going to speed up a little bit.
Okay, yeah, 0.6.
More wood, yeah.
Thought it felt like we all of a sudden hit a school zone or something.
Someone's getting bigger leather than his tools for opening course.
See a picture of it or something?
You sent a video?
Oh, you sent a video.
Well, he was showing people that
Another tripod fish going by on the left. The bones that we saw earlier, we don't think that they're whale bones, but yeah, still in the process of identifying what it possibly could have been. And of course, those from the Caribbean are all too familiar with the sargassum seaweed that we're seeing around here, especially from 2011. That's when it really started to affect many of our beaches. So I'm always interested in seeing how organisms interact with sargassum down here in the deep. We've had a few instances. Ooh, what's this on the left? Yeah, it's a squid. Very. Yeah. Nice. Very vibrant red.
Stay with us for a while.
Yeah.
You want to pause the boat?
Bridge. Or enforcement.
You want to stop?
I think he's got some.
Is it Alex?
See that? His antenna sticking out of there.
Oh yeah, over there.
He's got like a shrimp or something.
Yeah, shrimp. Does look like he maybe had a shrimp at some point.
Brushed his teeth.
Dust cloud behind me.
Mhm.
Focus on that there, maybe.
Got a nice squid here. I don't know if we've seen the species on our expedition so far.
Is that trash behind it?
Yeah, it looks like trash right behind it. Yeah.
Is it really?
I don't know. It's too square to be natural.
Yeah, it's definitely not natural. Did we get lasers on it? Could we? Yeah. Yeah, perfect.
I think he's got a shrimp in there.
Yeah.
You're right, I mean, look at the antennas, he definitely has,
Yeah, there's a shrimp in there.
definitely. He doesn't run right now.
No, I don't blame him.
Of course.
Much
Trust.
Does it also look like?
Got some strange arms.
Yeah, like some of the tentacles are shorter than the others.
Or is it just all this muck? Not zoom, sorry, focus. It's just
Yeah,
all
there's a
muck
lot.
around it, right?
Mm-hmm.
It's all about appearances. Mood.
Yeah, right by the bottom.
Mm-hmm.
Yeah. Weird.
Can you turn on the lasers now?
Same thing that fish was doing.
Yeah, some of the tentacles are gone. Like chopped. I don't know.
Yeah, but a little growth back, like they have little tips.
Mm-hmm. You got pictures with lasers?
No.
Oh.
Lasers was
It was like
blue
towards the
and
bottom.
part wasn't even the color too.
I don't know.
She loves some of the tentacles.
I don't know how possible it might be, but
What's that called?
no, Ryan was trying to get some photos with lasers, but I don't know how possible it would be to get them on the shrimp itself, the squid itself. So
I could zoom out and try it.
That's good.
I feel like it's digital.
That's good enough. I'm seeing it. I'm seeing the lasers.
that's good.
Yeah.
Thank you. And then something on its head is just like.
Scale balloon or something. Just below it.
Great show.
Mm-hmm. Okay, that's it for me, but I'll now hand over to my colleague Zahir, who works at the UE Zoological Museum, but I'm sure he'll introduce himself properly. And I'll say thank you to Manny and Zach.
You guys good with this guy, or you want to keep hanging out?
Good afternoon, everyone. Zahir here. Happy to be with you.
I'll be taking you through the next few minutes of the dive. Yeah.
Go ahead, Orbit.
You have some trash we want to take a look at as well. Yeah. That's all.
Update waypoint, are you still moving at 0.6?
We're gonna look at some trash.
Yeah, we should come up to point two and then
Okay, good.
So once again, we have some anthropogenic inputs. Unfortunately, we just want to document some of our projects are actually looking at the different types of anthropogenic inputs that we find at the deep sea. Any idea what that is, guys? Some sort of plastic. What? Yeah.
That's good.
Yep. So we are about 700 meters down. Another cephalopod. It's interesting. You saw this one before?
That's the one we were just looking at.
No, this is a same type of slug, but this one has its arms
Oh,
intact.
that's what I mean, it's the same kind, not the same.
Hmm.
It's not Fred. It's cool though, right?
Yeah, pretty cool.
Where are these? Where on the? Come sit like that, you know? Big coral or wood behind it too.
A woodfall. Yeah, it looks to be another woodfall. But it's been down here a while. We have stuff growing on it already. Yeah. Pretty large. Yeah, that's a really large barnacle actually. Some smaller barnacles, crinoid. And it's got its some sargassum congregating around it as well. One of our postgraduate students, Mr. Junior Thomas, is actually working on woodfalls at the deep sea. Pretty large shrimp as well, red rock shrimp. We're at the upper continental slope, about 700 meters, as I said. It's also known as the bathyal zone, lying below the mesopelagic zone. Quite a bit of different diversity we're seeing as well, compared to some of our deeper sites and some of our shallower sites. Interesting area. to waypoint 3, right? So we're making our way to our third waypoint, which is just at the slope of a ridge. We'll be moving along the edge of the bottom of this ridge to waypoint 4, and then make our ascent to waypoint 5 on the top of the ridge. We've already done quite a bit of sampling. We've utilized the slurper quite heavily this morning. And thanks to all our usual supporters for tuning in. And if you haven't done so, please give the SOI channel a follow. It really does help us out a lot. I'm not sure if they'll let you control the ROV, but you'll certainly get to see some amazing imagery, not from this, only from this expedition, but from their previous expeditions and the next one that's coming up, well, in a flash, really. Couple of rat-tailed fishes off to the right. And the ever-present red rock shrimp. We're currently off the northeasterly tip of Tobago, making our way around just off the coast of Castara. For those of us in Castari, maybe you can see the vessel. At this time, I just want to shout out our leading institutions and contributing institutions. Our hosting organization, as you know, is the Schmidt Ocean Institute, SOI, which is and their amazing flagship vessel, the research vessel Falkor 2. SOI is a private, non-profit foundation dedicated to advancing oceanographic research and the sharing of ocean knowledge. And this expedition certainly would not have been possible without their valued support. So a shout out from the people of Trinidad and Tobago to SOI. Our participating institutions being led by Species, which is a non-profit NGO in Trinidad and Tobago that undertakes scientific research, education, and advocacy. Other institutions contributing to this expedition are the University of the West Indies, St. Augustine, particularly the Department of Life Sciences in the Faculty of Science and Technology. And a shout out to my colleagues there. The Harbor Branch Oceanographic Institute, Florida Atlantic University, USA. The University of Calgary, Canada. Dalhousie University, Canada. The National Museum of Natural Sciences, Spain. So we have a quite diverse group of supporters and scientists on board, as well as our other supporting institutions. Ocean Discovery League, USA.
1.5.
Up to 0.5.
Pure Ocean, France, as we get a close-up of that shrimp.
It's halfway from the bones to the cliff.
The Explorers Club, USA. Very interesting name. The Pew Charitable Trust, USA. And the Anthony N. Sabga Awards for Caribbean Excellence. Special shout out to them and to all of our contributing entities. The expedition also consists of a team of 25 scientists and our resident artists, Marco. 15 of these scientists are from Trinidad and Tobago. One of them is from Barbados, so 16 CARICOM nationals. There are other researchers who are based onshore in TNT, but who are not on the vessel itself with us. So we have a very diverse, as I said, we have a very diverse group of individuals. And we also have a diverse crew who hail from all parts of the world. The accents on board are quite interesting. Seeing a lot of sargassum, both on the surface and down at the bottom here. Have a sponge up in the distance. Special shout out to Christian Gray, who's visiting from the States. Indulging in our local cuisine. And if you would like some memorabilia from this expedition, our leading institution, Species, does have t-shirts for sale. You can check out their website to get some more information. But it's a lovely Deep Wonders t-shirt commemorating the first Trinidadian-led expedition in our waters, Trinidad and Tobago waters. Yeah. The ground is, the bentos is littered with sargassum, as our pilot is saying. A little bit different to what we've been seeing earlier, entering a heavier zone for sargassum falls. Fish doing some burrowing in the sand. Just take a closer look. Some fish species do create their own burrows where they lay eggs to be fertilized by the male. Inquisitive white shrimp. So we're just trying to wait for this turbidity to clear to see what's actually going on in this supposed burrow. Got some sargassum in the hole as well. There we go. There we go.
Wow.
Yeah.
Is he gonna fit in there?
He came out of there.
Ratfish, huh?
Yeah.
Some really cool imagery. And he's going back in.
Sure, that's his own. Barely fits, folks. I mean, look at this. Unless somebody else is in there.
Yep, we weren't just looking at a hole. Possible rat-tailed fish. Interesting. Yeah. Very cool. He's a big guy, isn't he? Oh, if you casted it right, yeah. Yeah, the other end of the hole. Maybe he's looking for something in there. That's pretty cool. And I'm joined today on our data logging station by Mr. Ryan Monette, and our watch lead is Professor Anna Metaxas.
Jordan, you can go back to point 6.
RV bridge increases by 6.
Copy this.
So as I said, we're just off on the northeast point of Tobago. A beautiful island. If you're thinking about coming to the Caribbean for some sun, sea and sand, maybe think about Tobago. We're ready and willing to have you. Numerous resorts, as well as many Airbnbs. Come rent a car, explore the island, explore our lovely beaches, take a trip out on our glass bottom boats to explore the reefs, or if you're a scuba diver, come and take a dive. We have some of the clearest water in the Caribbean, and a host of wondrous diversity. If you're thinking more ecotourism, we can also do that for you. You can explore Tobago's main ridge, or even in Trinidad, or Northern Range, Acerite Nature Center. We have reefs all throughout Tobago, as well as reefs in northeast Trinidad. And some of the most amazing food on the planet. And I dare say the best KFC in the world. We should have some roti. Some roti, some curry, some dal puri roti, maybe. It's amazing. But definitely try our KFC.
Different, huh?
Yeah, it's the best. I've had it all over the world, and Trinidad is the best. I think I Show Speed would support that. And I don't have shares in KFC, mind you. So we're just proceeding along to waypoint number 3, doing what we do, exploring our deep sea, hoping to catch glimpses of something extraordinary, but happy to see the typical diversity you would expect as well. Tripod fish. Yeah, tripod fish.
They're so small.
Yeah.
You see them, you think they're like big fish.
They have
I've seen the tall ones like this, on the other side. But
interesting fin adaptations that allow them to stand up on the bottom of the sea like that.
Current's changing directions.
Yes.
You never know what's around the next bend. You never know what's over the next mound, as someone is saying in the chat. It really is a extraordinary environment.
you ever do the weird little flat sand dollar cast from yesterday?
I think it's a quite the oyster,
Yeah.
but I don't know. That's just a fact.
Okay. Yeah.
So, this is our science about this with the age these
Oh, yeah.
you know, recent stuff that they could be interacting with the conservation and the people who think that date is a fact distracted from the date and the other things. So we were wondering if well,
The mood lighting, that's what I do every time. Every time.
we passed inspection of finished powders. Jim said it looked amazing. Literally,
guys are very capable listeners.
Well, I mean, we're not, it's clean, yeah,
Yeah.
She cleaned the mirror, everybody cleaned the mirrors, I don't know why everybody cleans the mirror in the room, but everybody cleans, I guess because it reflects. That's it.
Um, 3 weeks, 4 weeks in, and you found it. That's the secret.
We used to have someone who would fold the towels, like
Oh.
the little animal, like
Yeah,
fold it.
so what we do is we roll them.
You roll them.
We actually took the towels off the racks, folded them.
That's so funny. Huh. See, going to that amount of effort is more than 90%. Zach, why are you staying silent?
I'm just agreeing silently.
Just mopped it.
Yep, mop the carpet.
Mopped the carpet, but it was wet, so then we
I was wondering what that smell was.
Another tripod fish. And a shout out to one of our shore-based scientists, Dr. Anjani Ganesh from the IMA and from Species. Hi, Anjani.
Current still okay?
Yeah, it's changed directions. It's still okay. Currently.
Currently.
Do you think if we see any more of those
Oh,
batfish here?
Batfish, yeah. Batfishes have a special fin adaptation as well. Their fins tend to look like legs. They can walk, in inverted commas, they can walk across the bottom.
Reduce this point to
Almost looks like a tadpole. Just turning a little bit to get a better view of us.
Just the swim with me from the other day.
this is stuff of stuffed animal.
Is that a smile I'm seeing?
Say hello.
What are you looking at? Inside.
He's kind of flatter than I was. He looks Italian. No, look at the.
Well, hold on.
I'm just imagining. Imagining like a mustache.
Someone write that down. Jules said that.
Do you disagree?
Yeah. I do.
Really interesting.
So ugly, it's cute.
Yeah, that's it.
I love the coloration. It's got huge eyes.
Just keep, so if I'm here, you just kind of want them. Good enough. No sound. Shall we leave our buddy? Or-
Yep.
Yeah.
I think we're all right.
Hi, buddy.
Later, dude.
Got some lovely imagery there. And we're going to proceed on our quest to find our waypoint 3.
Thank you. Keep your own laser if you want.
Okay, good.
You can continue later. If we keep finding these wood falls, do you think we should let Woods Hole know? Or Harbor Branch?
A little squatty. That's a lobster. White shrimp. Looked like a lobster. Yeah, but it wasn't clear. Like transparent like the shrimp. It was more like a spot lobster, but a lobster looking lobster. Sea cucumber. Oh. Yeah, you never know what you're gonna see, and that's one of the attractive things about this expedition. Constantly revealing new things. We've set so many records on the dive, on previous dives, finding organisms not only new to science, we think, but certainly new to Trinidad and Tobago. So quite a few range extensions for some species. Quite a few tripod fish we're seeing down this way. Seems to be running in place.
No need to be surprised.
Quite a few shrimp. as we expect. And a shout out to Miss Laura Smith, one of our constant followers. Happy to have you on again, Laura.
I'll tell you what though, I think so far this dive I've seen one rock.
Yeah, we're getting to the cliff.
We think it's,
We hope it.
could
We
be.
know it's a cliff.
It is quite steep.
You do know?
Even without exaggeration on it. it's
Yeah,
quite steep.
because the multi-beam was showing very hard returns.
We'll see. There's another one, back up.
Yeah.
I'm not saying they're good cliffs, definitely.
Nope,
But it
guaranteed
definitely is a
stuff.
It's pretty steep.
Unless it has consolidated.
Yeah, but not like this.
I can only go to the fancy one.
This is 2 times
So we're seeing a lot of flat substrate right now, but as we get closer to waypoint 3, we expect the elevation to increase and for us to see some different kinds of relief and hopefully some different types of diversity. So, as my time draws near to a close with you, I just want to say a special thank you to all of our listeners who've had so many kind words to say about me and the expedition. We really appreciate your words. We hope we did a good job for you. And keep tuning in not only to the next few dives that we do on this expedition, but also to SOI's channel as they move forward, spreading their joy and their mission of ocean discovery around the world. I want to say a special shout out to my cousin Rabia, who's always been on the chat and always been glued to the screen. Salams, Rabs. And probably for the last time, from the midnight zone to your screen, thanks for diving deep with the cucumbers and corals today. This is Zahir signing off from the deep sea, where the pressure is high, but the thrill of discovery is even higher. I'm now going to pass you on now to my colleague, Ms. Charisse Passad, who will lead you through the next hour of our dive.
No problem. Good afternoon, everyone. Thank you to Zahir for that lovely narration. I have a big shoes to fill with that. So as we continue along this dive, we're actually about to begin getting cores done. Those sediment cores are when we insert those cylinders into the sediment, and we get, it's approximately 25 centimeters long. And we then subsection those into what we call horizons, and I will be working on those tonight when they come on board, and we will be looking to see what is living in that sediment. So in the first 0 to 2 centimeters, what was living in that, then next 2 to 5 and 5 to 10. So that's 3 cores done that way, and then the remaining 9 go to Dr. Lydia Nakana and her team, where they will look at the for microplastic pollution and stuff like that on their end. Okay, 50 meters from now is where is 50 meters from? Just put a general point, because we'll want to take calls about that.
Could give us a signal.
Yeah.
Sure.
So in about 50 meters we'll take our 12 cores. Because I think from 3 to 4 is when it will be transects. And it may get rocky, so we want to get the soft sediment ahead of time. I don't know if you guys would have seen the map of the dive site we're at today, but we are, I believe, very near to Giles Island, St. Giles Island, which is an amazing dive site for anyone who's ever dove in Tobago's waters. You get a lot of corals and sponges and just a big reef system over there, and it is a drift dive. It is one of my favorite dives I've ever done in Tobago. So I'm excited to see what the deeper locations have to offer, and we are heading towards a steeper location. So we're hoping for some form of structure and more corals within that. But before we get to those kinds of areas, we need to take our course in the softer sediment locations. So we'll be doing that soon. I almost describe it as almost a Jurassic scene, because when you get up to it, it's like really lush green, and there's all these massive frigates all over. And then we usually pull up in a boat near the bay area where it's a little less current, and you descend at that point, make sure your entire group is safely together, and then you begin that dive out into the current.
Right, Charlie.
About 50 meters now.
Yeah, go to the stop.
Yeah, we could do the quiz. Yeah, we could set down. Charlotte, we're going to begin with sediment first.
Say again? Cheers.
We're about to hit a mark, and then we're going to do all 7 cores then. So we'll set down all 12 cores. It doesn't have to be exactly, just saying. So I think,
This might make us a little heavy, just
yeah. Okay. We just don't want to get too close to that rocky area, because then it'll be harder to put down. But it's trying to get a bit closer to the point, just because it's a bit heavy.
Yeah, sure. As close as we can get before it turns.
Yeah, so they said they'll let me know when it starts picking up on the sonar, and then not scale worms on something else. Not
That's
I don't
easy to
know.
look.
Is it?
Is
Or
it?
is it a scale? Scale worm?
You wanna stop and look at this, man?
Yeah, we're gonna zoom in if we can.
Weird looking fella. We passed over a couple of them, but couldn't stop yet.
Should we get it?
We can?
I don't know how, because,
Probably
section's temporary, that's one section.
Probably suction. Yes, bet.
What is the mesh size in one?
Oh, that's fine, it will fit, but it's just that that's our only.
Yeah, it's the only thing remaining.
I don't think it's worth it.
No, we can just get close-ups of it and continue.
Yeah.
Okay. So we won't sample it, but we'll just get as many close-ups as we can. It's very pretty color.
What is it?
Is it a some kind of polychaete? Or is it?
It's weird. It's like a polychaete mixed with a jellyfish or something. Jellychaete.
What should we log it as?
Thank you for your help.
Yeah.
Okay.
Yeah.
Wait, what do we log in it as?
You haven't locked it yet?
Well, I have it up, just waiting to click off what it is.
Oh, polychaete.
Oh,
We're not gonna try and get it, right?
No,
No.
but thank you. If you want to get a bit further, just roll weight. But we definitely don't want to get too close to the rocks.
Well, I think we're not gonna move the boat, so we could just back up if it gets too rough.
Okay.
We got nothing on the sonar yet. Nothing but nothing.
Yeah.
It's close to the rocks. We get something from the rocks.
Oh, it is a polychaete, guys, for those wondering. Polynoid.
Paranoid.
Scale worm. Oh, yeah. How much elevation did we gain? Feels like it got steeper.
And supposedly, that next waypoint is 525. But the start of this dive was what, like 715? So
Yeah.
Yeah, so we're what, at 6?
We're at 585.
Yeah, I mean, it's been a very.
Go ahead, RV.
So I'm just a small move, 0.6.
0.2 to waypoint.
So just a little more information about the dive site that we are at. It is a continental slope in the UNESCO Man and the Biosphere Reserve here in Northeast Tobago. So that also encompasses the Tobago Main Ridge Forest Reserve, and that is actually the oldest legally protected tropical forest reserve in the world. I believe it was back in, let me not say the date before I say the wrong thing, but it is the oldest protected tropical reserve, and this area is the first time it's being explored. So it is an exciting time for Trinidad and Tobago to be seeing 1776. I was thinking 1700s, but I'm not sure the exact date. So thank you to Charlotte for providing the date. But yeah, 1776 was when this was designated as protected area. And it is a gorgeous location to also drive through if you get the option. I have also gone to the hummingbird reserve within there, and there's so many hummingbirds, and they will sit on your hand and stuff like that. So it's an amazing place to be, and now to see under water beyond scuba diving depths is also such a privilege and an exciting time. Okay, so we're just getting ready to. We're getting as close to waypoint 3 as possible, and then we're going to take our 12 sediment cores before it turns into a bit of a rockier area. This is to ensure that we don't get the ROV too heavy too quickly, but we are getting to the base of that slope soon.
Got some good news.
Rocks.
Doesn't look like we're gonna have to worry about running into rocks.
That's not good news.
Unless it's being blocked by the steepness here, right? Yeah, still got nothing.
There's another one of those polynoid worms. Very pretty pink color. It is a baby bat. It's so tiny. Oh my goodness.
It's so cute, I want to die.
Oh my goodness.
Should I park?
Park and take some close-ups of it. Anna? Is
Yes,
it-
for sure, stop.
That is adorable.
What's that?
And I can tell that's what's happening too. It's such a concerted effort.
Oh dear. We did. What was hurt? Offended? Hurt.
That is teeny tiny.
It's smaller than the polyp.
It is smaller than polychaete.
Oh, well, not nice. Not nice.
Save your suction sound.
Look like it's like orange or poly.
Yeah.
It,
Let's get both lasers up by him real quick. Oh my gosh.
It's just too cute.
it
He's
is.
got wonky eyes.
Oh,
He's got googly eyes.
look at him.
It does.
That's not real.
I think this is my favorite thing ever.
Guess I met one of my kids.
You know, something is exceptionally cute when everyone in the mission control has pulled out their phones to take a picture.
I like how, you know, the other one turned to look at us too.
Yeah.
Curious, Chris.
Did you get a picture with the lasers? Do you see scale? No, when we were like further out in it. It's so small. Okay.
He's like, it looks blurry.
It does look blurry.
We saw one swimming earlier, you know, running along the bottom, and it was very shimmery at that point. Oh, go down real quick.
Go down.
Yeah. Right there, where the glacier was.
There.
Singapore.
Yeah.
What is
that? Look at that.
That's what it looked like. But it didn't was like attached.
Yeah.
Hasn't seen a film?
Yeah.
I still have cilia though.
The benthic ctenophores do as well. The red ones that we've seen before, they have a long cilia. It's a bit sticky, and that's how they catch their, how they feed. If we can even see the lasers in comparison to it. Oh,
What, 3 centimeters?
yeah. So,
Look at those.
just a reminder, guys, these lasers are set at 10 centimeters apart. So, what we're looking at is teeny tiny. The little feet.
I like his little bristles.
And there goes little fishy. Okay. Thanks so much. Unfortunately, we will be saying bye to a little friend to continue along. It's a hard goodbye. And yes, that was a ctenophore as well that you saw earlier, that very transparent, but you also saw that luminescence coming off of it. Bye, little guy.
Probably true.
Move a little bit more directly towards me if you want.
Mm-hmm.
Sure. Yeah, just like rectal.
And the sonar isn't even picking up anything rocky. No.
Looks like a big squishy
Bridge elevators, small one from me.
Oh, wait, sorry, what was that?
If it's
that's
looking
small
like
one
it's staying
from me,
soft
so-
in there.
Okay.
Or do
yes.
you want to do cores first?
Well, yeah, so it looks soft.
Mm-hmm.
We're just going to be going along from 3 to 4, so we don't expect a big change in some. Looks pretty similar.
Yeah.
Okay, we're going along that depth. So, the other thing is that is that another one?
Oh, flatfish.
Flatfish.
Wow, he's about camouflaged, huh?
Oh yeah, we need to take. Wow, yeah, I need some video.
Yeah. So we'll set. Look, we can set down and get some close-ups.
Oh. Something that is going to change here. Just remember transition 3 to 4. No, what happened last time? It's the same thing. This distance is only 360
Yeah.
meters, but we want 500-meter transit. So what they're going to do is they're going to have to go,
Shoot past it.
yeah, basically just go
Okay.
along to 500.
Uh-huh.
And
Yeah.
so if it starts looking hard, then we may start to transit.
The course.
Whenever we start with transit, just know that let him start with like a 270.
Yeah, wait for the 100 mark.
Yeah.
Okay.
Okay.
Got it. Right, so we've got a little flatfish here, so we're just gonna take some imagery of that. It is very dusty. Did we get lasers in as well? Look at it. Got fixed? And I guess we could probably just, yep, exactly. Look at it moving. There's some there.
Oh, yeah.
Yeah. Poly what? Polychilidate.
Soda in there like that, right? It wasn't even breathing.
Saw one swimming earlier.
Is it? Yeah. It is a tongue twister. Polychilidate. It's a.
See their larvae a lot.
It is a type of benthic lobster, like crustacean. Interesting little guy. I think this is my first time seeing one of those.
When they, like, if they come out and swim around, they look very weird.
They look a little spindly. Okay, I think we're continue along. And then what we're gonna do when we start setting it up between 3 and 4, that's only 364 meters, but we're gonna go 500 meter transect, so we should have to come up for that.
Okay.
Yeah. So you can set up a 500 meter transect for that.
From 3 to 500 meters.
Yeah.
And the push goes up after the transect.
Potentially, because it doesn't seem like the sediment is changing. If it does seem like it is changing, then we would have to cut the transect and then do cores. But as of right now, it seems soft. Oh, what? Yeah, it's like every time we get up to go, there we got another one. Is that it?
No? Look, the fin's going at the bottom.
Just very, very, very severe.
What is this guy?
So shiny.
Very shiny.
Yeah, filming shiny fish.
Oh, it looks so cool. Zach, did you set up all these fish along the way so that you could stuff? What kind of guy is that? That's some big silvery eyes. Oh. Look at the mouth. He is trying to psych you out.
It's a caffeine fish.
It's revving up to kill.
Oh. Bye, guy.
Flatfish.
I see someone's asking what we saw earlier with respect to the benthic crustacean. It was a polychilidae. Is it?
Yep.
And is it,
You have to see him get his little arms out. They don't look skinny enough.
looks kind of different.
They got skinny arms.
Alpha?
Hello. How are we doing?
You came in time for a transact?
Yeah, let's do it.
So we're going up to the line, and then you're gonna go that way, so you'll probably have to crowd, or maybe even just turn a little bit. I think if you turn sideways, it's not gonna be good, because it's pretty steep, right? So, and what else? Gonna do cores at some point?
Yeah,
Especially
probably.
if it gets rocky?
Yeah. So essentially, right now we're doing a 500-meter transact. When we get to waypoint 3, continue along.
Not long, is that?
Yeah.
Why do you hate me? I'm joking, it's the same.
But yeah, we'll do a 500-meter transact. We think the sediment looks like it will be staying soft, but if it does change to something different and it starts getting rockier, we'll cut the transact to get off course. But we won't cut the transact at like a random point. We will cut it at the 100-meter mark, essentially. So you get a full something happening. Big hole. Zach, you're really making sure you don't make it to 3.
No. Alright, you ready?
Yep.
Two tripod fish. Yeah. So we're gonna set up to typical transact height, 1 meter above.
Current isn't much of that, but I guess it probably go up.
So what do you people think about?
Are we currently? Just let me know if the camera is angled right above like porch height. So when you go down, see the porch and just one tick up.
You know what?
This second.
You want to see the porch?
Yeah, just let me see the porch and then we'll go one tick up, ready? So then up, up, up until porch just goes out of view. Perfect. And then we maintain like a. Well, right now it says 2.2 meters and this seems close. So
This is different. It's precise now because of the
angle, yeah. So
You have the altimeter.
we'll just have to maintain it as best as we can then, because I can't say exact height based on this. I'll let you know if it's getting too high.
What is this? We're gonna cross.
We're going at the very base along to, well, it will shoot past waypoint 4.
One of
Yes.
your lasers is blocked by a push cord too at that angle. So you only have one laser. There you go.
Lasers on, and then we get to get lower if we can. Before we begin.
Just
cores? Do them now? Okay.
This is the perfect sample, we have to leave that in.
Do the cores?
Here and then run.
All right. Can we set down into cores then?
Just nervous
Yeah. Fair enough.
conditions.
Not transects cores. Where's my pen? Okay.
You're gonna hit?
Yeah,
You want any clothes on the incline or it's okay?
It's not much of an incline, is it?
Yeah.
But the core's gonna come out like this? No, the core's still gonna be flat.
It's still gonna be flat.
Well,
Yeah.
don't get the core's like this. Make it magic happen.
You got this one? All right. Okay, then. Cool.
Good.
All right, so what we're gonna do is begin putting in for cores, and then that's essentially all on the RV guys. They will put the cylinder down into the sediment. They keep it pretty straight going down with a little space at the top because we do want to see what's in that first 0 to 2 centimeters. So we try to keep some water at the very top of it, and they'll go directly in, pretty straight angle. Can we push down a little bit?
Huh? Can I?
You want to bring it up one at a time? Let me confirm.
It's up to you guys.
I think we might have to push that down a little bit further, but not too much. We still want some gap at the top, but let me confirm. Can we
Normal is to do.
Yeah. Yeah, we can push down that a little bit deeper, so we just have a space up to about
We're gonna know.
here.
Okay. Are you doing one at a time, Claude?
Huh?
One at a time and pull back.
Yeah, you will.
Yeah.
It's faster now, we.
Yes.
According to
Yeah, no,
you
that
guys have
works.
time.
That works.
Right there. So we still get some water at the top. Perfect. Right. That's core 8. Coming back in. It is a bit angled. No, I think
No.
it's 590 is the next one.
It's push for 8.
And it's push for 8, yeah.
Oh,
So sample ID is 590. Perfect.
Sample is the sample.
Can we try and get a more level in core? The sediment can get a bit more level. It was a bit angled. I know where. I just had a.
So if I go down straight, it's not gonna. See there, you want me to push it in at an angle, or you want me to push it down straight?
Straight.
So the top is gonna be at an angle.
Yeah, the first call came up with a bit of an angle. That's okay. Yeah.
Well, that's fine. We'll start it here. So that's fine. The course should go in straight.
Okay.
Not-
We'll keep it. This thing?
No,
What you're showing me? Five nine. Which core number is that? 7? Push score 7, sample 591. It always looks like it's gone in as deep as possible and then. Let's push score 12. Push score 12, 592. Probably next week we'll be putting it a bit deeper because there's a bit more water. Push score 11. See? Is that straight? Nice. And that was 11, right? That was push core 11?
Yeah.
Yeah. Perfect. The trip? No, tomorrow. There's 3 dives at this side. There's supposed to be 4, but that first night dive couldn't happen. How can you guys see? Oh. Got this. Push core 10? Yeah. If possible, maybe we don't take any more samples where the porches come out because I think it's disturbed that sediment. Because we look at those first layers especially. So. Zach set you guys up. He made sure to stop at every animal he saw on the way to doing this because he did.
Yeah, that's the perfect one.
Five, 9, 4, push core 10.
Sorry.
Look at the brittle slaw go. Trying to get away from us. No. I think Alex said the same thing about the little batfish from earlier. He was, when I pointed out the brittle slaw just now, he was like, do the push gore, and I was like, no. And then I remembered you with the bat.
Could have got him in there.
Oh. Deep. Yeah. Last goal of the trip. That was 6. 595 for sure. Square 5. It's a bit too deep. Five, 9, 6, push for 5. Next one. No, so we have a little gap. Right, there's the. There's the. I said right there's the. It was a nice cake. I still can't tell what flavor it was though.
Banana.
When I said it the first time, no one believed. Everyone.
How was the food? Was the food good?
The what?
The lunch was good?
I had no idea. I just had the cake.
I had the
Was
fish.
the food good?
I just had fish
It
and
was steak,
cauliflower.
steak and eggs.
Steak and eggs?
Steak, eggs, fish, cauliflower.
The cauliflower was really nice.
It's not rice and eggs. It's good. Dinner's rotation.
Agent.
Yeah.
What core number was that? 9?
9.
Fish core 9. 597 fish core 9. Four more. Yeah, the 4. In the quiver side. It's like, would that be a problem? The sediment gonna fall into it? Yeah.
Done?
Wait, what? We didn't do those.
These are all good now, so it's only the This one was that.
No, yeah. I'm confused. Is it?
Well, hasn't been done.
No. None of them should have been done. We picked up 2 and it looks like there's sediment in it. But
I think they marked that on one, that's one.
yeah, I think we might have to. No, no, no. That was coral quivers. Yeah, there was stuff in the coral quivers. There should be nothing in the sediment core. Is it possible that when we that pushed out, things went in? But to that extent, no, because the base is sealed. Yeah, we pulled it out and then the top of it was full and they put it back down confused. Yeah, we'll shake it out for sure. But we'll do 4. Down. Oh. show you on mine.
Right. And then, so this is wrong.
So that two is actually this one.
So this should
Yeah. Yeah, your torch map is cool. Four, just going here. 598. This is one. There's a transaction between three and four. And when you do this, is between three and four, so it's a 5 year transaction.
Cool.
Yep. All right. While we call this
What do you want to jump across?
if you can get this guy in first and then move, if that works. Because yeah, I think beyond them being buried, they're gonna have to get shaken out.
Which one?
I think it was 2, not the one currently in your hand. That can get put in, but remember when we pulled the push cord 2, there was stuff in it, so that'll get helped.
With an 8.
Yeah.
Huh?
The one with an 8 you can
All right, guys, so I'm going to hand over to Robert, who will be with you for the next hour. Thank you for joining in.
Yeah. Hey everyone, Robert Cassidy taking over while we continue looking at dust. Doing some push cores right now to get some sediment samples. We actually have a lot of samples today because we found a bone fall, which we think was from a swordfish or something like that. Kind of a fairly large sized fish. So we have a lot of samples from that area.
Side here, because the push cores are forward.
Sorry, I don't understand what you mean. Yeah.
Push cores are under the sun there. I want to jump subtly.
Yeah. So they're under the sub, like you can't get them.
See the push cores there?
Yeah.
The first tube.
Oh, I see.
Forward.
Oh, yeah. Okay. Yeah, yeah. If you can back up a bit, that's great. Yeah, I couldn't see it with the dust.
Well, this too.
That is pretty soft. So.
Well, they just put it in. They just put it in. So number 4 is what you've collected, right?
Yeah. 1, 2, 3, 4, yes. Oh, I just retracted it around so far.
Yeah. I mean, if we can get to it.
This position.
Yeah, so for these ones, I think maybe Sharice told you already, but we want to try to shake them off, get the sediment off before we put them in.
We don't want to let sediment landing in the water.
Yep. Wow, yeah.
That's a lot of sediment.
Yeah, is it just a forgotten cord? I don't understand how it's in there. You want to zoom on it? Yeah, because you hold it up to the screen actually, just to see.
Yeah.
I mean,
clear.
it's good now. Yeah. Cool. Thanks. Yeah, we want some space above, right? That's good. Okay. Great. Yeah, so you can see we're dealing with some pretty thick sediment here as we get these cores. Can make things challenging visibility-wise, but these cores are really important because they allow us to sample the fauna that's actually in the sediment. It's just the kind of thing that you can't see any other way with video or anything like that. So it'll give us an idea of the kind of things that live in the top layer of these areas that we're in now. And we are right at the edge of a pretty steep slope of sediment here as well. So it's possible that the conditions change as we move along the waypoints. Got a fish in the top right corner just kind of watching the whole process, and he's off. There we go.
You guys don't mind waiting, right?
What's that?
Don't mind waiting. Our next option is you can like try to come up easy, put it in, come back down, and do the three like that.
Because what's the problem?
The idea was that you would push the sediment away by coming back down the thruster. Yeah. As opposed to waiting for it to clear.
Or if you
for
go
a long
to a
time.
different location.
We don't want to be far. They want all the cores in the same area, so.
Okay.
Yeah,
There's nothing much we could do because
I know.
we just tried it with having like the least amount of thrust, and it's like
Will it matter if you turn into the current?
if thing
Let's
is,
stay in
how
the
we are
same.
that incline,
Yeah, we
we're
can.
gonna be thrusting
Yeah.
into the walls. It's kind of,
Yeah.
this is the best way for us to like sit on the incline.
Well, it seems to be like slowly clearing enough.
Yeah,
No?
so we can.
Yeah. Have you taken the core or are you just hovering?
Yeah, I'll be in my hand.
It's just not in the quiver yet.
Hmm?
It's just not in the quiver yet, but it's
But
in
it's
the
in
box.
there.
Yeah.
In the core. Yeah.
Yeah.
And I would just have another twist.
What is that?
What's that one?
Consider lifting and landing somewhere else, but not very far. I don't know how far we can get. How close we can stay.
Well, I mean, we could try what they were saying, which I think was just moving forward a bit and then going back to blow away the sediment.
Yeah, sure.
Yeah. So the idea is that we would like, if we want to try to blow the sediment away with the thrusters, we would move forward and then move back, or how would that work exactly?
So that again?
You were saying before that we could like blow the sediment away with the thrusters, right? No.
Yeah, right now we can pull it, but
We would move far away from where we are now, or we would stay kind of close still. What do you think?
yeah, we could try. So my method was not even sitting because sediment is so soft, so kind of come up, right where we could see it like now. Put
Yeah,
it
now
in,
you can see it. Yeah.
Right.
and put it in,
Yeah.
and then go back down.
And take two more.
Yeah.
Oh, yeah.
More.
Sure.
That's
yeah,
what you were saying
sure,
basically.
Yeah.
Okay.
Sure.
We could try that.
Okay.
Yeah. I mean, otherwise we'll just
We're here all
be
day.
watching
Basically.
mud all day.
Yeah.
Yeah, but go outside.
You know,
Zach, on the Prusa there, if you want to cancel to use, you can cancel.
No, I don't want to cancel. I was just curious what it is.
But for that, they ask for store some plastic pieces to go in.
Alright. That's it. So
There you go.
So you have number two.
Yeah. Or just we're inside the slope. That was 599, right? It's not as exciting as the bone fall, is it?
No. That was intense.
Yeah. I mean, I walked in and saw. I didn't realize we had gotten so many samples from it.
Yeah.
It's cool though.
Well, because we weren't allowed to use the pores, so they like took like 2 or 3
Yeah.
samples around and then went into the gate and they sent them out.
Yeah, it's cool though.
So now we don't have unsanctioned samples and use it wisely. We use it wisely.
Yeah, I mean,
So everything has to happen.
That's what here we're doing the course. So like, if you see one,
Yeah.
where we take it.
Yeah.
I think there's a lot of like Ocedax worms on the bones. Are
Cracked.
huge.
Cracked.
It's cool.
Yeah, that didn't even test sick.
I don't know.
Each little worm had an isotope on it.
Right. That's cool. I wonder if they have Ocedax collections from this area. Probably not. But that's cool. Yeah. My supervisor does.
Everybody was so excited.
Yeah.
And certainly not as excited.
Yeah. I mean, it's crazy.
Crazy.
Yeah.
Okay. Good. It's good.
There we go. Yeah.
So when we're done here, we're doing a transact, and they've already laid it out. So if I
They've already laid it out?
They've already laid out.
Nice.
So let's start.
Okay.
Ooh. Cabin. What about 45-degree angle?
It's a bad day to be a sieve.
These things are.
Definitely not gonna wash it. I'll just put it out of damping and move it so it's kind of middle of the bell. So it's not hitting the side at least. What's crazy is that's only just the top.
Yeah.
Like the floats off in that shit. It's only,
Yeah.
it's just rolling up the fire. So we're not moving.
Something pretty.
It wasn't like that earlier when we were at 7 o'clock. So I wonder if it's because the float's going on or something. You have to watch it really close when you start cutting that board, you know. I wish there were 3 that aren't here for the dog. At least when we move, yeah, we'll be moving with the current.
Just don't want to go too deep down. You could leave like that much of the clear plastic. A little bit more. Oh. That's okay.
Okay, everybody's very sorry about this. Okay, that's our last one, right?
Yeah.
Perfect.
Is that enough? I mean, it should be
we'll
right.
have to-
Yeah.
Yeah. All right.
Awesome. Very nice work, guys. Thank you. So when you're ready, we can head towards that transect and start.
So, has the current switched directions?
sideways or forward?
Want to go forward? I mean, it's gonna be on a slope, right? So the points will be different distance apart. You want to go sideways?
How much of a slope is it? Is it really
At
that slope?
45 degrees.
What?
Like this is 1x exaggeration.
Wow.
I mean, we're in early days on it, but yeah, it's a.
So, shall we do it sideways?
I think that's better, right? If we can.
No, I can put the camera up a little bit. You gonna try?
Yeah, if you want. Or you good with your
Alright, good with camera.
Looks like it's
I was pointing, huh?
Pointing
Before?
out now?
No, before. Now
So it's good now?
Yeah.
Yeah, so we just want to flash the lights when we start the transects.
Up there, Wakeman.
Start.
Oh, she usually likes to see the shadow. What? It's time?
Got to have an evaluation.
What?
From Zach.
Alright. I'm not gonna complain. It's gonna be different. Sideways.
Thanks, Ref.
The altimeter is on. Well, I don't know if it helps me much. Because we are not here.
How you doing?
Good, how are you?
Good. Yeah, so we're just in this transect. Going sideways, like Ref said.
So we want to do the transect sideways, facing it.
Yeah, and we already started it, so we can just keep going. Yeah, sideways to the right.
started? Has this been noted?
Yeah.
Did you note that we're doing this sideways? In case the observer can see it when it's raining. So there's no way you guys can go forward there.
Yeah, I'm just slowly going on it.
Yeah, you guys can get it forward. Well, won't the points be off if the slope is so steep?
Oh, it's out rock.
Oh wow. Yeah, so we've got some whip coral here on these rocks.
Camera anymore, right? So
I'm not really
I
sure
can't see stuff.
when I'm advising.
No?
It's gonna be very difficult because you can't see none of the.
Yeah.
You guys wanted to
Well,
follow it?
how do you feel about transit under this condition?
Not good because I've not got a camera facing starboard, so I'm kind of blind.
Good. So let's
Mm.
just forget about the transect. We can't do the conditions, but
Okay.
so we'll just go forward and
Take a look around.
take a look at it. Yes.
Okay.
There's a sun on it, see?
Yeah, so we were doing a transect here. The conditions are pretty tricky though, given that we're on a very steep slope and there's lots of sediment. So we're actually going to abandon that and just kind of explore, fly around, see what we see. We're seeing a much rockier area now than we were before. So hopefully we'll get some different animal communities, as we're starting to see here with these coral. Very cool spiraled whip coral sticking off the rock. So we're really up against like a 45-degree slope right now, so it's actually pretty dramatic.
You didn't mention me. I was working towards the summer now. Okay, maybe see if the last evaluation.
Yeah, lots of coral.
So you want to kind of stay at the base of this as we transit across.
I think we want to stay at the base of this, or do you want to kind of go up and down? What do you think?
Yeah, sure. We will be.
Maybe some like.
Whatever works. I mean, I know it's tricky flying, so.
Yeah, I mean, it could be interesting to see what we get further up. Like, we're kind of right at the very bottom of.
But I think the point is we'll go to 4 and then we go up.
And then we go up? Okay.
Yeah, so.
Okay, I'll stay as low as I can.
Yeah,
If you see
and it's
anything.
Okay.
yeah.
Yeah, we'll let you
Yeah.
know. Thanks. Looks like.
It's not beer, it's not beer. Beer gets all excited.
Yeah, of course.
We can go up here.
Yeah. These crinoids down here?
Wow, yeah,
Huge, huge crinoids. That's. Some of those look like they're about 20 centimeters wingspan, I guess. I don't know if you say wingspan for a crinoid. They're on, yeah. Pretty cool, huh? We got interesting fish down here, too. Oh, yeah. You seeing. Okay. What do you want to zoom on? Okay. When you can, Manny, if we could get a zoom on. Show you some of these crinoids here. No rush if you're not stable yet. Actually, yeah, like down here would be a great spot to get a good zoom and focus on. I can see these crinoids really like at the lip of this rock formation, presumably because that's the best spot for them to grab nutrients from the water. Wow. Look at that. And one of these sponges, I think, that we've been seeing also. I think we got a few of those a couple days ago. Wow. Is that? It looks like a big sea spider. Also, didn't even see it first. So much, just suddenly, so much life here. All of a sudden, you know. Yeah, it's what, when you get a hard substrate, and you get movement of water. That's what you find, really. Wow, this is a big sea spider. I can tell that there's probably all kinds of stuff living underneath these lips of the rock too, like these fish. Wow. Beautiful. Oh yeah. I forget what kind of sponge that is. Faria, I think maybe is what Shirley said. Wow. Can we zoom up here too? I think there's some more sponges in that nook. Yeah. Yep. Right there too in the middle of the screen. Yeah. Oh yeah. Very nice. Cool. I think that's good there. So we could pan out, maybe take another look to the right. So we could kind of like move along, yeah, this area a little bit. Here? Yeah. That's a black coral. Wow. I don't know. It's pretty big though, isn't it? It's is that what they were calling a redfish? Maybe? Or, big fish down here as well. Not sure what kind. Wow, cool anemone here. Would you mind just zooming on that for a sec too? Seeing quite a few different types of coral here. Got these white ones, the orange whip corals. Maybe. Yep.
There was something yellow
Below us.
here.
Oh yeah. This thing? That looks like. Is that a coral, you think? Yeah? Wow. I think you can actually see a small cup coral inside of that little hole. Wow. white one? We can check it out. The big camera.
What's now,
It looks like another one of those white corals. Maybe. Another big cluster of crinoids here. More this kind of orange yellow coral, and that cool big white one here too. Ooh. It'd be cool to zoom. Yeah, that one. Could be. Yeah. So these crinoids, even though they look a lot like corals, they're actually closely related to sea stars and sea cucumbers. It's a totally different phylum, Echinodermata. Can we zoom on this one down here too? Yeah. Well, I gotta choose a sponge first, right?
We can have a sponge bias here, just because you're at the sea.
Looks like a sponge is. The crinoid is growing on the sponge though, which is cool. Really cool. These sponges are really brittle. They kind of crumble. They're really not spongy feeling at all when we've sampled them before. They've got this very cool, kind of semi-translucent tissue. It almost looks like plastic when you hold it up close. Yeah, that'd be great. You think Chrysogorgia? I don't know. Hmm. Cup coral there too, actually. Really small one. Yeah. Beautiful. All right. I guess we keep heading this way now. There's probably a lot more to see. Yeah, just look at the slope on this wall here. It's almost a vertical wall. Another anemone, more corals and feather stars. Looks like a white sponge there. Big sea stars. Oh wow. Got another cool, very circular, spherical sponge here. Big, brittle star. Wow. Yeah. Could you zoom on that for a sec, Nanny? If you can zoom any more. That's pretty cool. It's like a tennis ball. Yeah. Wow. Cool little lobster right here. Type thing. Lobster? Don't know. I'm not sure what it is. Huge shrimp or a giant squall lobster? Something else. Yeah. It looks kind of rigid to me. Alright. Gonna continue onward as we explore this very cool wall that we found ourselves at. Looks like a different kind of coral down here, maybe. Oh yeah. Big, pretty speedy crab. Oh, he's tangled in the coral. And what is that swimming right there? That's the crinoid taking off? Wow. Can we follow that for a sec? Is that possible? Look at that. See, you don't really expect it when you're just looking at them planted on the rock there, do you? But you can do that. Wow. Just looks like an alien, doesn't it? Wow. That is cool. Just parachuting down. Wow.
Of course, helping you out there.
Wow.
Whenever you're happy, let's switch early and then you can switch back early because they want to be little Charlie.
Okay, you want in?
Whenever you're happy.
It's pretty amazing as well.
Yeah.
That's pretty cool. You got here at the right time.
You haven't
Yeah.
seen any flat spots, have you? Not really.
That's somewhere to land.
Just for little Charlie.
Yeah, there's quite a few ledges, I think.
Okay.
You want to do the flyby? Yes,
Yeah.
we'll be up there.
Yeah, it'll be flyby time.
Yeah.
If we can get someone to set her down.
There you go.
I mean, I don't think we're gonna have too much of a. It might look good. It might look like fog lights or something.
Yeah. I'm just wondering exactly whether we'll get there as we're going up from 4 to 5. Up the slope there. to get away from all the parts more. I mean, I don't know, just guessing. I guess it's 3 years.
I don't really have a clue, to be honest.
Yeah, we've got a bit there. Hard chance.
Okay.
And that's late season.
I saw the sea spider too, that's cool.
Very cool.
Sea cool. Picture down there. You can come back early, so I don't rip you off.
Hey, doing, Zach?
Good, buddy.
Pretty good. On a cool wall right now.
I mean, yeah. I don't know. I guess either way is fine, right? I figure I'll go up, I'll put the rams out, get up against it, and you can grab it so it's real steady, and then just, it's a loose handle, so if you set it down, we should be able to tell if it's going to fall or not. Have y'all seen any especially big, like corals or anything?
Nothing huge so far, I don't think.
What's corals? So we like just got to the rock time?
Yeah, we didn't move much.
This is steep I'm looking at, right? That's not flat.
Here's like this.
Like going up there? Oh, straight up the gully?
That's too high.
I don't know. I was looking like, see right above that whip coral, I think there's a little bit of like right in that kind of neighborhood. Might be worth a try. You could get it in your hand and just not let go, but just put it on there and see what happens, you know?
Hi everyone. Thank you for those who are joining us. I will be narrating for you for the next hour. My name is Eugenia Thomas, and I'm a marine biologist and MPhil student at UE St. Augustine, studying deep sea biology.
Would we be able to
And-
put these into a file box? EB3? Or that must be outside?
Zip ties, if you break it up.
I mean, we have.
Samples are in there.
No.
Well, I would put it with fish bones. I know that's super special. What about an empty one of these?
Yeah.
That'd be okay?
Three, yeah. Yeah, it's okay. He put the zip ties hanging out in one of the Bible boxes.
Can we put
So,
the zip
zip
ties in
ties.
the empty one?
Yeah.
That's okay?
For a small fur of amount of money.
How about 50 cents?
Yeah, look good. They fell all the time or it's this time?
No, they just like pop down in a way where they're like right here.
Okay.
You know,
Yeah. Oh yeah, I get it. Cool.
Okay. Open 3.
So we are exploring the northeast side of the UNESCO Man of the Biosphere. This is our 8th site, site 8B.
Nice. What's that? Okay. Are you trying to straighten that or wait till that's close them? Much better. Lasers coming on. Bunch of thermometers.
So we are continuing our exploration on the northeast side of the island of Tobago. And we've seen some pretty interesting organisms. For this dive, we've seen a lot of coral species, and we've also seen we also see a fish fall. So a lot of that oceanographic worms. So those are keystone species usually on whale falls, but it was quite interesting seeing it on fish bones. We believe that it's swordfish or some other type of fish, but it is pretty interesting seeing oceanic worms on other species as well, other than wheelfalls. And so for Site 8, we are mainly exploring the mesophotic reefs. And this zone is typically between 50 to 150 meters. See a lot of octocorals and starfish in this area. We also saw a squid this morning. I don't know if you all were wrong for that, but we saw a pretty cool squid. To lots of crinoids. What? Yeah, can you give me what's that?
Which one do you want to see?
This.
The anemone looking for?
Yeah. Oh yeah, I think it is a sea anemone. Yeah, that's a sea anemone. Yeah? Yeah, just closing, yeah. So, right here in this frame, we have a sea anemone that's just retracted itself into its internal body. So usually you see the jelly-like organs are outside of their body, but now it's internal. So it's pretty cool to actually see it in this form. We're actually seeing more corals today than our dive yesterday. See a lot of associated organisms on some coral branches. Seeing brittle star, some shrimps. This is a nice one here. And one of the reasons why there is a possibility that there are more corals today than previous dive is because this dive have a lot of rocky outcrops, and they are known to attach themselves onto the rock where they They recruit themselves and grow into full-grown adults. And see a lot of different corals now. See sea anemones. This one is pretty cool. So we are commonly seeing the orange octocorals, but we are now starting to see more of the white ones. See some fish as well. Oh, this one is beautiful. See a small squat lobster. And they are also known to be hanging off of the coral branches. Just taking in all the organic matters or any small microscopic organisms that may drift with the currents. So these squat lobsters are really opportunistic species. There's a bigger one here on the seafloor. Actually, yeah. Yeah, so they can be on the seafloor or hanging off of coral branches. This one is really nice. Don't think we see much of these. And these lobsters are known to use their antennas to help navigate throughout the environment. And this is a interesting fish. Yeah. This fish is sort of hiding from us, but this is the colors on this is almost like a ombre. Seeing a bit of purple. Yeah, oh, this is nice. And look at that lateral line. Beautiful. It's sort of hiding itself within caves. So we have our... Yeah, oh, gee. Yeah. Yeah. So we have our fish expert, Farina Solomon, on this expedition with us. And she's saying that is a G. Darwini. Oh, they want.
You can say the common name. Darwin's slime head.
So the common name for that fish is the Darwin slimehead, and the scientific name is the G. darwini. And our expert, Dr. Farinas, is also saying that it is a commercial fish species. They collect it in Charlottesville, which is also along the northeast side of Tobago. And she's saying they also call it the ugly fish. I guess our locals calls it that. So that was pretty nice and interesting to see. Could you zoom in on the thing? Yeah. Could be a very large polychaetes. What is that? I don't know if it's a polychaete or is a like probably a detached arm. Like that. Yeah, so I mean we're not sure if it's it looks like a polychaete, the body structure it resembles that. Or it could be a detached arm. So so it's more than likely a polychaete, a flatworm. Right? It's a very interesting color. And this will be our first time we've seen this type. You know, polychaetes are commonly found in all marine environments and at all depths within the water column. And interestingly enough, this particular species has no face, so it could have bioreceptors that they use to help them navigate within the seafloor. Pretty interesting shrimp. We've seen a lot of these species. This is one of our common species we've seen throughout the dive.
Here, close. Where? No, it's like that. No, I'm not sure if maybe the other one's the worm.
The worm? Oh.
It's not a polychaete. This might be a nemertine or
Nematarian.
Yeah, they were saying I think good job there.
can you make some more zoom in on it?
Zoom
It's
in?
just underneath us, right?
No, it's like that. But when I park, I can get super deep paper seeds.
Okay.
There's no real, and it's nice when there's stuff.
Bridge arc.
Yeah.
There's one.
There's one there,
Yeah, maybe with the slurp.
So we're going to investigate this species a bit more. Definitely haven't seen it on any of our dives, so
We cannot slurp them.
Oh no.
We don't have space for slurp.
That's true.
You have
We
to
have
Yeah.
only one number one, but
Only
Yeah.
if you keep it at number one. It doesn't
it has to be something big. No, but I think
seeing a small so we're just really getting some close-up zooms of this organism here. Oh, wow! So it's burying itself almost like an earthworm. You know how they bury themselves in the sediments. So this is definitely longer than what we I've anticipated. And if you look really close, you could see some of the organs in its body structure as well.
No, John is not in this channel. Only you, Sean. And I think they're filming right now. Sure. I think they're about to film, so he might be. There. Okay.
So it could be a pneumatia. So yeah. So that was pretty interesting. Yeah, looks like a sponge. Interesting color.
We are landing. Let's try to zoom into one and see how it looks.
Yeah.
Can you
Zoom in on
these corals?
the corals.
Sure, which one?
The orange
Which
ones.
ones?
What corals?
Yeah. We have limited time, so maybe on these ones, yeah.
On those?
There's
Oh yeah.
this
those are
guy
nice.
there, where the starfish is no, is it a crab?
The brittle star?
Yeah.
Yeah. You want some to go back?
That's actually pretty good. Maybe, yeah. Probably something like that.
So we're just
Is there a different, without picking the light that is taking too much time out of the view, is there a different light that we can?
Oh, we can certainly play with them. He's a bit off to the
Here, you see, this is destiny.
So we're just investigating some of the whip corals.
Maybe
A bit more, we've seen some pretty cool brittle star on it.
Crazy Dr. Seuss garden, yeah?
What?
Dr. Seuss garden.
Yeah.
It's really amazing how we are seeing so much deep sea corals at only 521 meters below the surface of the water. So we're gonna play around with the lights, try to get some cool footage. So previously what we were looking at was what we assume it is a pneumatian. And we actually saw it burying itself within the sediments. And so these are known as borers. They either bury themselves in the sediment to hide from predators or to feed on any organic particles, matter that are in the sediments. So really getting some cool close-up footage of these whip corals. And corals are very, very, very amazing species. They have hundreds and thousands of individual polyps that just makes up this entire colony. And then so many other organisms attach themselves to the structure. There's also a very cool squat lobster as well. This little guy is just here hanging around. Oh, looks cute. And so he's literally, hanging around feeding on organic matter within the water column. And of course, as we've all seen throughout these dives, they can also be found on the seafloor. So again, these are mostly opportunistic species.
Go darker.
So it's amazing how much light, different light intensity we can get from the ROVs to get close-up images, get some cinematic images as well. It's really nice looking and just observing their behavior. Oh, this is a background here. I think we can see a crab. But this is a beautiful brittle star. And you look at the tube feet that they use to help them move along the seafloor and up coral structures. And so similar to other species in the Echinoderm family, they all have tube feet. It's really nice to see what different lights can do.
The last one there, and then we need to move.
Yeah, we need to move because we are at the decline.
Less than 30 seconds, Marco.
We need to move, guys. Sorry.
Yeah.
Okay?
Yep.
Let's go.
We need to go. Yeah.
All right, let's go.
All right.
All right. Ladies back.
Yep.
Sir.
Rich Arthur. Hello, you can continue that move for point one. Yeah.
So we're going to resume exploring the continental slope, northeast side of the UNESCO Man of the Biosphere. Let's see what other interesting species we can see. Hopefully some new ones. see a fish. Tails almost look sort of translucent. Almost looks like those needlefish. Very cool. Yeah, so it's still also, it looks translucent and okay, it's hiding from us. Yeah, we can go.
Do I hear we're collecting that coral or?
Yes, the white one.
Yeah, the white one.
Yeah. And before we collect it, can we get like a shot?
So we're going to.
Doesn't matter which one, right, Dean?
Yeah, no.
Either one.
Whichever is easy.
So we're going to collect a coral sample. And what's so nice about collecting these species is that you don't only collect, you know, just the coral, but you collect other small organisms that are on it as well. So you can also collect some amphipods. What are we seeing here? Yeah, we're seeing some amphipods on it. Yeah.
Can we have a poll in the picture?
Again, we're playing with the lights. See what type of different cinematic images we can collect before we sample this beautiful and amazing coral species.
Like that?
So guys, thank you for tuning in. It was nice chatting with you for the past hour, and now I'm going to pass you over to Megan, who will be narrating for you for the next hour. Thanks guys, have a great day or night.
Is this going in a bio box?
Yeah.
Bio box?
Yeah, Biobox 3. That's basically all we collected. Water. All this is, yeah. And we can always do it point.
Yeah. But this one I prefer in the Biobox because they are like too fresh.
Yeah.
El blanco Biobox because it's porque es muy ramoso.
Yeah, just like that.
Hello, my name is Megan Conkling. I'm a research scientist at Harbor Branch Oceanographic Institute at Florida Atlantic University in the United States, and we're here exploring